It's 9 o'clock, so let's take our seats and then get Eric can help get us started. Hey Eric, can I control the slides up here? Great. Oh. So we'll just wait a minute while we get the webinar started. We'll get a countdown. Can't go backwards. Are we started? Oh, excellent. Um, well, good morning. Welcome. It's fantastic that we're finally at this point where the Chemical Sciences Roundtable at the National Academy of Sciences could come together to close out a year on artificial intelligence topics and meet in DC on a cold February morning. So in this room and attending virtually, we have a great group of researchers who understand that there is an opportunity in science to change lives. This is where AI is becoming a game changer, and this is the topic of this workshop. AI-driven drug discovery is not just about speeding up research. It is the goal of this workshop to focus on personalizing medicine at scale. So what if we approach every illness as if it were a rare disease? You know, if we could tailor treatments with the same precision given to orphan diseases, healthcare disparities, they could begin to disappear. No patient would be left waiting simply because their biology doesn't fit into the mold of existing treatments. This idea and topic of drug development has been part of my life on several occasions. As an undergraduate, I got to work on bioengineering methods to produce vinblastine and vincristine from Cantharanthus roseus, better known as periwinkle plants. And that was for the treatment of juvenile and leukemia. Years later, a close friend of mine had her graduate studies interrupted by ovarian cancer, and she told me it was cisplatin that ultimately saved her life. And cisplatin, just such a rather simple and elegant molecule. So with this in mind, I'm excited to kick off this workshop. For the next day and a half, we'll explore how AI is transforming drug discovery, the challenges with bias and data, and how we have the opportunity to deal with them. There's an underlying theme with this group of scientists on laboratory automation and robotics so that we can realize this idea of, like what I mentioned earlier, personalized medicine at scale. And we'll hear from outsiders of the world of AI, where citizen scientists have banded together to solve big problems, like searching for new treatments for cystic fibrosis, using access to the internet to teach millions of school kids to computer programming, and NASA's discovery of exoplanets. So I look forward to engaging discussions, and let's get started. Just some housekeeping if you're online. I think many of the CSR members are online. Please use the raise hand feature if you're going to ask questions. And Slido can also be used by the general audience for asking questions, and the staff here will help filter through those questions and give them to the panelists. Just an event disclaimer. It's essential to the National Academy's mission in providing evidence-based advice that participants in any of our meetings or events avoid political or partisan statements or commentary and or commentary and maintain a culture of mutual respect. The statements and presentations during our meetings or events are solely those of the individual participants and do not necessarily represent the views of the other participants or the National Academy of Sciences, which is a nonpartisan tax-exempt organization that includes under its charter the National Academy of Engineering, National Academy of Medicine, and that operates the National Research Council. We have a policy that prevents discrimination, harassment, bullying for participants. All forms of discrimination, harassment, and bullying are prohibited in the National Academy's activities. This policy applies to all participants in all settings and locations in which the National Academy's work and activities are conducted, including committee meetings, workshops, conferences, and workshops such as this. I just said workshops twice. So let's get started. I'll kind of go through these so we can get the ball rolling. We have 3 panel discussions on AI and drug discovery and a breakout foresight activity that for participants, for the CSR members and the participants in person in DC. All of these are on the agenda and available on the agenda online. The folks that help, here's a list of everyone who, I don't want to go through all these names because it'll take forever, but I'll just call out the planners that helped out. The co-chairs of the Chemical Sciences Roundtable are Nikki Pohl and Marty Burke. Linda Chin, Carlos Gonzalez, Sammy Coello, Laurel Royer helped organize these, help organize the meeting and pull all this together. For their fantastic list of panelists are up there on the right. These are our roundtable members. We have kind of 2 sets of roundtable members. We have roundtable members, and we have ex officio roundtable members. Those are people based on their position, mainly in the United States government, that are offered seats on the roundtable. For that reason, roundtable activities at the National Academies have an inherent conflict, shall we say. So we cannot provide recommendations to the US government. But we can have these amazing meetings where we can come together, have these discussions, and pull together. We'll have a proceedings and brief that will be published, capturing all the conversations we have that will be sent to the United States government and available online to the wider community. I never introduced myself. I'm Michael Janicki. In the back, helping out, we have Darlene Kay, and Apoorva is going to help out with a foresight activity. Eric is running the meeting, and so he's going to make us all look good by pinning us exactly the right moment. And there's a lot of other staff in the building that are going to help out with this meeting. Just a couple things. The proceedings and brief from the last meeting have been published. You can go to our website and download them. You can also use this QR code. And that topic was on publishing in the age of open science, workshop for researchers, institutions, and publishers. And this meeting is being recorded. So if you miss things, you can go back and watch the recordings. So many little things you forget when you have these big meetings to talk about. The Chemical Sciences Roundtable is looking to fill some missing seats at the table. So if you're interested, please go to our website and either nominate somebody, or you can also nominate yourself for consideration. This is the culmination of a year of topics on AI. So we did AI and sustainability, AI and scalability, AI and limited data. The recorded meetings are all available on our website. This year, the Chemical Sciences Roundtable have decided on the topic of a forward-looking topic of what AI in 2050. So we're a quarter way through this century, and what will we look like in another quarter century? How do we get there? What are the challenges? What do we need to do now to sort of meet some lofty goals? And what will chemistry look like in 25 years? With that, I don't have anything left on the other opening remarks. I'm looking at Marty and Nikki. They'll have remarks later. Excellent. So let's move into the first panel. So the first panel, Andrew White will introduce the panelists, and we have Alexandra, James, Nicolo, and Jesse Kirkpatrick to discuss drug discovery now. Like, where are we with AI? some success stories, some challenges, and just some real life examples of what's going on. You might have to. We only have 4 chairs. So would the panelists like to come up and sit at our nice, comfy, synthetic leather chairs that were designed by chemists? Naga hide, perhaps? Huh? Okay. Yeah, they just make it. Okay. Yeah. So each one. Okay. Okay.
Oh, okay. Hi, everybody. I'm Andrew White. I'm an associate professor of chemical engineering at University of Rochester, and I've been on sabbatical for about 1 year at an AI bio nonprofit called Future House, which I co-founded in San Francisco. I'm happy to be participating or leading this panel. And so the topic of the first panel will be drug discovery, the future is now. And we're going to discuss artificial intelligence, machine learning, robotics, and automation, and how they're affecting the progress of drug discovery. So our first panelist is Dr. Alexandra Snyder from Generate Biomedicine. And I think we're going to have each speaker, each panelist spend a few minutes talking about their background. And so please go ahead, tell us about yourself.
Great, thank you. And do you want me to do the AI, my thoughts on AI as well, or are we going to do that next?
Yeah, I think if you want to spend a minute about your thoughts on AI, that sounds great.
Great. So it's an honor to be here, and I look forward to the conversations over the course of the day. My name is Alex Snyder, and I am the head of research and development at Generate Biomedicines. We use AI and ML to create biologics. So a few things that I wanted to touch upon sort of at a high level before we dive into the discussion later. I wanted to talk a little bit about how I came into AI, the framework that we use internally for thinking about. Is that good? Good? Does anyone else hear that? Okay. Do you hear the crackling? All right, I'll just keep talking, and if anyone feels like they're going to have a seizure, I'll stop. So I'll talk a little bit about my path into AI-based drug development, how we're using a framework to think about conceiving of if we're making progress and measuring that progress, what our experience has been so far with developing drugs, and in particular interacting with regulators, and then just touching upon the risks. That's a reality that we all, I think, need to think about. So I actually came into AI not via anything computational. I am a simple medical oncologist with no computational training at all. What I did have was a sense of intense frustration at the quality of the medicines that we're developing. Now, of course, there have been some that have been transformative, but there are too many oncology patients who are still suffering and dying from cancer. And what I felt was that really we were developing the drugs that we had rather than the drugs that we wished that we had. And so in starting to investigate AI-based drug development, I started to see some data that I felt was promising. Now, this took time. When I was initially in large pharma after leaving academia, it was a time where there were lots of buzzwords being used, huge promises being made, which just didn't seem credible. There's a little bit of that still going on, but I think overall we're moving into a place where people are speaking with more precision about what we expect these tools to do, what their limitations are, and what their applications are. So in that vein, when the framework that we use internally to think about how to measure what AI is doing in drug development is that the probability of success of a medicine helping people is the product of right target or targets, right medicine, and the right clinical experiment to adjudicate if that medicine is helping. And that you have to get all 3 of those things right to know if AI is contributing in a positive way. Now, at the moment, as we all are aware, there are different investigators and companies working in each of those three spaces. Many are focused on just one. You know, there are some that are really focused on identifying new targets or multiple targets, really dissecting disease pathology in ways that haven't been done before. There are others, including us, who are really working on the right drug problem, that middle part of the product. And then still others who are developing tools to, for example, identify the right patients across the country or across the world when you're trying to accrue a study, using AI to collect and handle the data, even to write CSRs, things like that. So there are all sorts of applications that are progressing, I think, diversely in the US and around the world. And I think for a plug, when we are looking at the news that's constantly coming out in this space, we encourage folks to think about when you know, one of these headlines come out, AI has failed its promise, or, you know, these sorts of things that we read. Well, let's dig deeper. What was the hypothesis? What was the AI doing? If the AI was trying to do right drug, but the wrong target was chosen, then let's talk about that. So we're really encouraging folks in this space to try and dig deeper into applications, just as we would with any other scientific hypothesis. I think for folks who aren't in the AI field, it's sort of a big, scary black box, whereas we're trying to encourage the dialogue to be more specific as we would with any other scientific tool. In terms of our experience, we've now had 2 drugs that were designed using the assistance of AI go through phase 1, and a third is about to start. So we've had interactions with various regulatory authorities, and we've put in very thorough IND packages to make it very clear that we are not trying to cut corners. We are not suggesting that AI enables us to skip steps of evaluating preclinical predictors of most of all safety as well as efficacy. And so we found that it's been really quite smooth sailing in that sense. So finally, let me go to the risk piece. I think as with any new and powerful technology, there will be applications that have constructive intent and destructive intent. And of course, we're trying to be constructive in making medicines and programming biology. But just as we are making proteins that are intended to be medicines, there's no reason why someone with destructive intent couldn't do the same thing to make toxins or to engineer viruses, et cetera. So our feeling is that it's our responsibility as a community to be introspective, to regulate ourselves to the extent that we can, and to participate in organizations, especially public-private partnerships, that are trying to figure out how to make sure that these technologies are used for good and try and protect the world from contexts in which they may not be. So let me pause there and pass to the next speaker.
All right, thank you. So our next speaker is Dr. James Lee, who is president of Gondola Bio and has a PhD from Stanford in biochemistry. Thanks for joining us, and please love to hear from you in your opening remarks.
Yeah, Andrew, thanks for the introduction. Very excited to be here, and very excited to be learning from the workshop participants and panelists. So as Andrew introduced, I'm a biochemist by training. I lead a company called Gondola Bio. We're a Bay Area biotech company focused on developing therapeutic for genetic disease with high unmet need. And the company was created last year as an R&D spin-off of BridgeBio. And BridgeBio has a similar vision, has been around for a little longer for various corporate and financial reasons. We feel it's a better way to serve the patients by splitting the company into 2, where then we can more creatively and broadly target more innovations for rare disease patients. I'm going to provide a little bit of background on the company and this vision, and then we'll talk about a couple of case studies and observations that we're seeing on how AI has been helping advance the therapeutics development here. BridgeBio was founded in 2015 with a vision that at that time, and even nowadays, people say there's more than 7,000 different diseases. And a lot of focus for the therapeutic development field has been the 5% or 500 diseases that are target a pretty broad population, and that affects like millions of patients. And that's great. It really helped a lot of patients there. But there's almost like 6,500 disease that no one really touches. There's no approved therapies for. And in many cases, there's actually not even a pipeline product for. And while each disease may represent a small portion, it could be a couple hundred patients, could be a couple of thousand or tens of thousands. So individually, they may not be as impactful of the disease of millions. Collectively, they are affecting tens of millions of Americans and more globally, and it creates a substantial disease burden. So the vision of the company is like, what's the best way to tackle this disease at scale? And it's not only about doing 1, 3, or 5 disease, but it's about, can we do 10? Can we do 30? Can we do 50? So over the course of the past 10 years, we've bought close to 30 pipeline products, 19 INDs and 3 NDAs. And we continue to work with a lot of academics and other industry partners to see, can we even expand the number? Can we go faster? So we have a little bit of a different model of the company that we call hub and spoke model, which is essentially a decentralized R&D model where we put all the assets and all the research into individual affiliates. And each individual affiliates is almost a minimal viable company that focus on one specific disease or one specific molecule. And its sole mission is to advance this as quickly as we can, use whatever methods. Many of them would include incorporation of AI tool to accelerate the research and development. But then we do have the central platform in earlier case, BridgeBio, but now BridgeBio and Gondola Bio, with the sense that we provide the scientific guidance, some of the central logistic and operation support so that the company can function at scale. And the other part of this, we tend to invest in uncorrelated area. The investment thesis of the company is about genetic disease, whether we have human genetic validation and whether we're targeting disease as a source. So our pipeline is quite distributed, which diversifies the risk in the drug development. And such diversification not only give us the opportunity to serve more patients, but also provide additional avenues of financing. So again, we can do more programs here. So because of the decentralized fashion, it's almost each individual program has the opportunity to make their own decisions. And we have then the opportunity to experiment different AI tools. And I want to mention a couple of the visions here. The first one is not about the chemistry per se, but it's about finding the right targets. It's important because the most important thing in drug development is getting the right target. If you're getting the wrong target or the wrong approach, it doesn't matter how good your tech is. It doesn't matter how good your team is. The probability of success is essentially zero. So what we've done and what we've seen the field done is by integrating a lot of genetics and more broadly omics database to interrogate 2 questions. One is, are there human genetic evidence that are in terms of allelic series, in terms of different mutation impact human traits that would suggest modulating the target would be disease modifying? And the second question we ask is also by modulating the target and or the pathway, are there liabilities that we need to be aware of, either early in initiating the project or monitoring throughout the development, depending on what liability we're talking about. So our computational genetics team has spent a lot of time in integrating the data and use these machine learning algorithms to find out what are, to tease out these large data set. We're not necessarily use AI to generate insights human cannot predict. We hope we have more of that, and sometimes the insight does surprise us. But in most other cases, what we find is AI can do like 80–90% of the job, but it can get done very fast. It's like if I have questions this morning, I can ask the team and they will get an answer in an hour or two was a preliminary answer. But if you have to dug into the literature and do all the research, it could be taking days and weeks. So it's really a tool acceleration, give you early answer, give you early hypothesis that you can pursue. And then after you get to the right right target and the right project, obviously the key question people ask is like, where's my chemistry starting point? How do I advance my chemistry starting point? And how do I de-risk my chemicals? And this is where we've seen both in our research ecosystem, but also with various partners we have where AI has been playing a critical role. Just a couple of examples here, and we can go into the details later. One is about virtual screening. And the challenge about HTS is, of course, you can do it high speed. If you have the library, if you have the experience, if you have millions to invest, you can get to the target and blast through it. The challenge for biotech like us and for many academics, they have access to neither of those resources. So these in silico tools, and which are getting better and better and becoming more available, you can run a pipeline and generate a couple of chemicals or like 50, 100 chemicals that it will be interesting in testing within a matter of days and within a matter of weeks. And you don't actually need wide experimental tools. You can then validate with protein, with cell system, with certain synthesized chemicals. But again, it lowers the barrier, increases the diversity you can test, and enables small research team to conduct work that used to be 50 people, 100 people, or requires multiple partners. Another application we've seen is once you get that hit or once you advance into the lead, how do you iterate much faster? And here is like using computational power to drive the SAR and using computational power to predict the pharmacokinetic properties of the drug to gain certain PK parameter, for example. We've had a lot of success in working with partners like Lawrence National Labs and with certain AI providers. So it's not our chemists then don't need to think about new chemistry anymore, but they tend to have, let's say, each generation of data, they have like 50, 100 ideas. This allows them to vast like, oh, this 5 or 10, we should really prioritize and let me put the resources on this. And they enable the faster turnaround and iterations. Because each round of synthesis of this chemistry take a while. It could be like 20 and it could be 5 to 15 steps or sometimes 20 steps. It could take weeks. So how do you reduce iteration you need? How do you get higher chance of success? And using a lot of the predictive tools, using AI algorithm, they can tell you, oh, this design have red flags, so you should avoid that. Or this would improve oral bioavailability, you should try that. And another aspect we've seen, speaking of the synthesis, is it's also not trivial, right? A lot of times think about which are the synthetic routes, which are the catalysts to use. And we've seen a lot of the AI tools being able to offer good suggestions, again, within a matter of minutes and hours, that you can speed up the synthesis by selecting the right route. You can try more creative ideas or parallel ways so that, again, instead of taking weeks, you could reduce the timeline. And the most expensive thing in research and early development is time. And patients are waiting. There's a lot of unmet needs. So the faster we can go there, the more value we could be creating. And we've also been using AI in like looking at different phenotype predictions, trying to figure out what disease people have, and in terms of trial recruitment. In the interest of time, I'm not going to go into a lot of that in more detail. But what we're seeing a general theme is it's not a substitution of what the team is doing, but it really provides the tools that enables any individual to be really trying things out and generate a lot of new ideas. Even last week, I had the opportunity to meet with 2 PIs, one in London, one in Hopkins, that that actually presented very interesting ideas. They're physicians by training. They have interest in biological targets. And generally, if it were like 5, 10 years ago when we go out and meet them, they would say, hey, look, it's like, I have this exciting target. I have some ways to think about it, but this is just my idea. Like, you guys need to try it out. And that could take years. That leads to a lot of questions. But what they both of them independently presented is like, oh, I use this like AlphaFold, Rosetta, or whatever tool. I designed some tool peptide that actually I inject into the mouse. And now I can show you some mouse data. Or I have this co-crystal structure that can then run a screen for small molecules. So this really, and it's done by labs. Even again, 5, 10 years ago, wouldn't have any capability of generating starter points. So it lowers the barrier for a lot of researchers and lower the barrier to target smaller diseases. So hopefully, overall, we can benefit more patients.
All right, thank you. The next member of the panel is Nikoli Fusi, who's a general manager at Microsoft Research in Cambridge and has a PhD in computer science from University of Sheffield. So thanks for joining us and love to hear your opening remarks.
Thank you. Okay, can you hear me? Okay. Very nice to be here. Thank you so much for the invitation. It's a great event. So yes, I'll specify Cambridge, Massachusetts. We also have a lab in Cambridge, UK. There is competition, but we always like to specify. Mostly because we are in Boston in a great ecosystem also, so good to say. So I'm at MSR. So MSR is the research arm of Microsoft, and we've been doing something in biology at least for the last 15 years at different levels of intensity. I joined Microsoft Research 11 years ago as a statistical geneticist. I mean, my background is more in machine learning, but through the connections between Gaussian processes, which I was working on, and linear mixed models, that's how I ended up in genetics. And then Microsoft Research happened to have a research group near UCLA in LA working on genome-wide association studies. And that's how I started. And the interest around biology and the life sciences at Microsoft has only been growing, and it has really reached a kind of like a step function change, I would say around 5 years ago. And in terms of just like amount of people and resources spent on kind of trying to figure out models of biology. A focus on my team in particular is on scientific discovery and automating scientific discovery as much as possible, as much as practical with generative models. And we have a little bit of a pointed view on this because we think, so there is this current in AI right now that basically says you can learn a lot of things about nature just by querying the model itself. And that brings you pretty far. Like if you look at the result that came out yesterday from Google, you can do some discovery up to a point. But ultimately, the scientific process involves some sort of interaction with nature, particularly in the life sciences. So at some point, you need to do an experiment. And that's where a lot of our focus is. We basically look at that from a little bit of an information theoretic view, and we think that there are only two things you can do to make the scientific discovery faster and better, process faster and better. One is you can accelerate that loop. So every time you have a scientific hypothesis and you have to query nature to figure out if that thing is real or not, you can design an experiment, you can perform it, you observe the result, and then you can speed up that loop at the rate at which you can perform experiments. Or you can try to draw more information out from nature per loop. And that involves new lab techniques, new experimental techniques, better experimental design, better systems like biological systems, and so on. And so that's where a lot of our focus is. So to do that, to both accelerate the loop and to get more bits of information per interaction with nature, we are designing models that I use this cheesy, this is a very cheesy sentence, but like they learn to speak the language of nature, meaning they can understand and produce the readouts that we expect directly in the right language. For instance, we can design protein sequences, we can design protein structures. we can design DNA sequence or small molecules and design the system that they go into as well. So that's what I mean by speaking the language of nature. So the idea is, can you come up with a model, like a GPT class model, that not only can understand natural language, but can also design a protein pretty well in context or in as part of its thinking loop. And I know that Andrew has been, I follow your Twitter a lot with your experiments with molecules, and recently Grok, I think you tried out.
It failed, by the way.
I know. That's which makes me happy every time I see that something like that fails, because it's kind of our point. You kind of need some specialized training to do that. So and going back to speaking the language of nature, one of the things that I feel like a lot of the bio community or the comp bio community hasn't fully absorbed, I think, is the bitter lesson that Rich Sutton first proposed, which is very well absorbed by the AI community, which is general methods that leverage computation better tend to do better in the long run than super specialized methods that have been very carefully crafted. And it's a super bitter lesson, because I built a career out of building super fancy things with a lot of prior knowledge. But in some sense, what we're seeing now is that if you build a general purpose method and you select your data carefully and you just let it train, it will do a lot better in the long run. And so that's unfortunately our focus now. And so just, I don't want to take too much time, but like there are a few themes that I see emerging from our work and our experience in the area. One of them is that as a field, folks working in AI biology, we tend to say that we need more data. Like, every conference I've been to in the last at least 10 years, maybe 8 years, maybe with Gen AI, it has even exploded. We keep saying we need more data. We need more cells from our single cell studies. This is a theme that I think we need to kind of open up and inspect a little bit more carefully. We don't need more data. We need better data. We need less of it. So we have a couple of papers. One is already out. One is going to come out this week in single cells, showing that you can throw 1 billion cells at a problem in single cell RNA-seq modeling, foundation models of single cell RNA-seq. But if you just dump all of the data into the model, it will not learn anything. And in fact, you could learn on 1% of the data and have a better model. And so the second paper shows that there are particular ways to design how you gather that data to do a better job. So this meme of we need more data, we need to kind of like, yes, we need more data, but the right kind, and that's going to take a while for the community to figure out. The second big theme is we work on generative models. And one of the things that we notice working with the community and collaborators is that they're very hard to use. People really do not yet grasp a lot of the nuances and intricacies of working with a true generative model. And so sometimes I give them this example, which is, imagine that you could get a file out of the sequencer, but instead of coming out of the sequencer, it comes from a generative model. And that tends to open up some kind of different thinking, but people still think of generative models as very fancy databases that you can retrieve knowledge from. And instead, like, they are kind of like machines that give you hypothetical words. And so or simulators, let's call them. And so that's how we use them to accelerate the loop. They simulate outcomes in nature. And when they're very uncertain, we go back and do an experiment. But that kind of usage is still not very widespread in the community. And it's interesting to note why. And as a result of that, I think use cases for generative AI in biology are still kind of very few. And people tend to use these models as they use the foundation models of the previous era. So I feel like there are kind of 3 main eras in the field of AI and biology. The first era was everybody has their own data set, everybody trains their own model on their data set, and that's their favorite model. And I'm going to use this model for all my experiments going forward. Then there was a second era, which I think started maybe in 2015 and maybe ended with AlphaFold 1, where people really said, let's train fewer models that we're going to call them foundation models, and we're going to fine tune them on our data. And that's going to be my favorite model now. And then I feel like we're entering this third era where we have, in the limit, perhaps one model class. So like a GPT style model class. But we are going to do a lot of our thinking in context, so in the prompt, so to speak, of the model. So there is no weights. No weights are getting thrown around and changed. But that induces new use cases that people kind of have to think about. Anyway, I'll stop here, but thank you.
Great, thank you. And our last panelist is Dr. Jesse Kirkpatrick, who's a research associate professor at George Mason's College of Engineering, and he's the co-director of the Mason Autonomy and Robotics Center. Thank you.
Great, thank you. Good morning, everyone, and thank you for the invitation to be at this event. It's wonderful. It's an honor and a privilege to be here today. So I was asked to speak about three things. First, I was asked to speak about my story, which I'll tell you a little bit about. I was asked to also speak about self-driving labs and ethics. So I'm going to try to do justice to all these three things in the course of 15 minutes. All right, so first my story, which I think is, each of our stories are unique, as we've heard today, in our paths to where we've gone and how we've gotten there. But I think that there are some general things, key enablers, that are really important when we think about our own stories, that are important for scientific research, and this research area in particular. So my story is somewhat interesting and unique in the sense that I'm in the College of Engineering and Computing, as was mentioned. I also co-direct an Autonomy and Robotics Center. My background is actually in social science and sort of like secretly more in philosophy and ethics. So how'd I get to where I am really quickly? So I did a PhD in political science at University of Maryland. And while doing that PhD, I was offered the opportunity to do some work at Johns Hopkins Applied Physics Lab. This is kind of like my introduction to technical work on the national security side, which was super interesting. From there, I went and did a postdoc at the U.S. Naval Academy, continuing some of that technical work and also this kind of national security theme, right? And it really kind of oriented me towards thinking about the relationship between policy, ethics, and technology, particularly with a technical focus. And that's kind of carried me throughout my career, right? So I've worked only in public institutions, but I've done a lot of consulting work for the U.S. government and also for private industry. And so while the focus today is on a range of topics, one is particular emphasis on AI, I want to talk about responsible AI. And that's something that I work on a lot. I work on in my current position. And so, right, so what is responsible AI? What's the relationship to drug discovery, to my story, and then into ethics? So responsible AI, as many of you probably or likely do know, is behaving responsibly, creating AI responsibly, innovating responsibly across the life cycle of a system. So design, development, deployment, and use. Right? I think all of the speakers have already kind of touched upon these elements and some of the work and some of the considerations that are consistent with this work, right? And so there are these kind of general principles that there's deep disagreement about, but right, that we hear a lot about trustworthy AI or safe or secure AI, and these may seem like these may seem like sort of conceptual or without teeth or non-technical. But I think when the rubber hits the road, and we'll see this, I think, when we think about drug discovery, is that when the rubber hits the road, if you want to actually operationalize responsible AI, think about doing AI responsibly within the context of drug discovery, for example, then you need to start to think about what does it take to operationalize? So what does it mean for a system or for AI in the context of drug discovery to be trustworthy, right? So For a system to be trustworthy, does it need to be secure? Does it need to be safe? Does it need to be reliable, governable, whatever the case may be? And then how do we build that into the pipeline of our workflow, right? And in addition, how do we measure it? Right? So AI metrology is a huge part, and that's like how we measure certain fundamental characteristics of AI, certain benchmarks. But how do we know that it is, behaving, that we're designing it responsibly is going to be contingent upon how we measure it, what we hold ourselves up to. So really, a lot of the work that I do is thinking about this in the context of my own individual research and specifically in robotics, and then thinking about how we design systems specific to responsible AI and how we build them at scale across organizations, right? And that's really, I think, fundamental to what we think about when we think about the research enterprise more generally, right? So what are some key enablers to doing that I think are specific to my story, but also important when we think about the scientific enterprise more generally? So I think first is interdisciplinary thinking and partnerships, right? We've heard the various disciplines that are, and we'll be hearing about the various disciplines at play within this field and within this existing and emerging area of research, right? It takes all types. Could talk more about that later. I think deep stakeholder engagement, right? So knowing what people need and listening to them. I think the other is bridging AI capabilities and ethical considerations. I talked a little bit about this in the context of artificial intelligence and responsible AI, but I think that one thing is considering the broader impacts of our research. That's what many of us want to do when we think about the research that we're doing when it comes to drug discovery, public health. And having this be part of what we consider to be the intellectual merit of our projects, right? So thinking about the broader implication of our work. I think it's also crucial to have, as a key enabler, stable, predictable funding sources, right? That support and facilitate basic scientific research, okay, across the board. And science free of partisan politics is crucial, right? To be able to allow scientists and researchers, no matter where they are, to be able to do the work that they want to do. And that is important to moving the ball forward for the well-being and the good of us all. All right, self-driving labs. So as many of you know, and I think that this will be interesting to hear who might be using self-driving labs, or I'd say self-driving labs, or the idea here is to integrate AI, robotics, and automation for experimental workflows. I'll just say a few things, right? So why they matter, and then what some of the kind of bottlenecks are currently, and then what some things that we want to think about with relationship to these are. So, right, they help in AI augmented experimentation. And we've heard about, high throughput screening or automating workflows with AI, right? You can think of adding a robotics platform on top of this. I'll talk about why there might be some issues with that. Right, breakthroughs in drug discovery and material sciences. There are some really interesting papers on nanofilms and how workflows have been improved with the combination of automation and self-driving labs with respect to robotics. Really interesting is potential sustainability and efficiency gains. I think that's pretty crucial, right? So optimizing and reducing chemical use by reusing reagents, really kind of cool applications there. Minimizing the number of failed experiments, right? Potentially reducing the amount of animals required to be used in experiments and potentially reducing suffering on that end, right? Which I think is important to us. Okay, so what are some limitations and challenges, right? Most of them are at the sort of robotics level, right? You'll hear lots of people kind of pump and hype robotics and sort of their job to do that, but there are some major limitations, okay? Most self-driving labs currently operate on or rely on pre-programmed workflows, okay? So they're not autonomous, despite what you may be told. Robots can pipette, they can mix, they can measure, but they struggle with novel and unexpected situations, simply like issues like clogged syringes or hardware failures, right? They need humans to go in and fix them, humans in the loop, so to speak. There are some or many flexible robotic arms and soft robotics in early development, but they're not yet widespread, right? So there are hardware limitations here is my general point. An important one that I think we'll hear throughout the day are just simply data limitations, right? Limitations of AI models, right? The type of data that we have, from whom it's drawn, who it represents. Is it diverse data? Is it representative? Is it, and those are all going to play a role in the efficacy of self-driving labs and AI design in general. All right, cost and accessibility is another one. Super expensive, right? It's the big players that are going to be able to use self-driving labs and to design and develop them, and then they're really tough to scale at industrial level. So primarily in the research phase. All right, so I think I talked about some of the issues. One I think is really interesting and I hope that gets to be on the agenda today is the kind of open science versus proprietary models, particularly when it comes to AI. And I think this is a big debate if we're thinking about artificial intelligence, which is, is it going to be commercial AI, commercial proprietary applications, or open source? And there's been some really interesting developments over the past few weeks that have, the jury's still out, right? Maybe it'll be a mix of these two, but those are going to have direct implications when it comes to global access to drugs and to drug discovery and who's involved in this enterprise. Similarly, who develops these innovations, right? I don't need to remind us, but I will, of kind of our Sputnik moment where the federal government created DARPA, NASA, the National Defense Education Act, right? And in which the US government recognized and prioritized that staying ahead in science and technology was a matter of national security, global competitiveness. Whether that will continue or we'll see a fundamental reorientation wherein staying ahead in science is primarily basic scientific research is wedded tightly to commercial applications and takes place in the private space. All right, so in closing, how are we going to tackle key questions related to the integration of AI and robotics when we think about drug discovery? Right. I think really imperative here is, and this is like sort of full circle part of my story, is equipping researchers with the knowledge, skills, capabilities, abilities to consider and advance the broader impacts of their research, to really think about the societal implications and give them the space, the opportunity, the means to do so. So again, the three things: interdisciplinary thinking, partnerships, stable, predictable funding, sources that support and facilitate basic scientific research, having an environment that is conducive to doing so. So with that, I thank you for your time and your attention, allowing me to tell my story, what I think are some key enablers for it, and some critical ethical issues that face us that are going to be really important as we think about science going forward. Thanks.
Thanks. So we have a few minutes left, and I wanted to take a little bit of time to learn about how everyone found their way into AI, because I think it's a, kind of a niche field, maybe when we were all doing our professional training or academic training. And I think everyone has kind of gotten to this point in a different journey. And I think it's really interesting. So maybe, Alex, we could start with you. Why don't you tell us a little bit about like how you were able to get up to speed on, generative protein models, which I think is not a topic 10 years ago or 20 years ago, and how you stay up to date on it.
So I'm not going to claim that I am perfectly up to speed on generative AI models for protein design. As I mentioned, I came at this from a very different angle. So medical oncology training, I've been in biotech and large pharma, where these things were really not integrated into the fabric of what we did at that time. I think things are now changing. What pulled me into it was actually wet lab data. So to some points that have been made before, computational data on its own, in our view of looking at what the data shows us, is absolutely insufficient to get to the best possible drug swiftly. One needs, at a minimum, computational data and wet lab data, and where possible, structural data accelerates and refines even more. To your exact question of how we stay up to speed, I'm fortunate to be surrounded by people who are deeply expert in far more than myself, folks like Gabor Gregorian, who's our founder, John Ingram, who's you know, a real leader in the field, and we are constantly self-educating. I think the point that was made earlier about where all this is going in terms of using publicly available tools, creating your own tools, publicly available data, proprietary data, I totally agree that the jury is still out. The approach we're taking right now is to try to stay up to speed with what's publicly available, and we're generating a lot of our own wet lab and structural data to try and just continuously elevate the models. But I think one of the things I love about this community is that there is actually a lot of discussion and publication going on. And we have had, as was referenced, some moments lately where we've said, oh, wow, some of our most core assumptions are shaking at their foundations. But all right, let's listen to that and adjust accordingly.
Great, thanks. James?
Okay, this is on. I firstly, I saw them coming here to get up to speed. I'm not, definitely not. So, so the way I get into this AI and drug discovery thing, I think is a little bit more organic. As Alex mentioned, it's not necessarily, there's always a lot, all sorts of hype. It can do this and it can do that. But what kind of bubbled up to us is, hey, like this academics or this research lab or this scientist in our laboratory has been using this tool so that he or she is faster on getting this experiment done, or it takes less iterations, or it generating some interesting leads that has good animal data, and later it has good clinical data. And that's where it gets attention. And I think it's a lot more organic in a sense. The way this tool works is make you more efficient. It gets you more creativity, and those ultimately translate into a wet lab data, and you can do it at lower cost. And then because ultimately the data speaks and the science speaks, where would you see kind of what's the latest. We also don't claim to be at the cutting edge of it, but rather what we do is we look at the literature, we talk to the experts, and we try to just being able to adapt what is the latest and what is the best. It's like, for example, there's this new generative model or new data set that other people have used. Most times it's not being used for the exact disease and applications we're using here. So what we've done a lot is integrating those into our system so that we can answer specific questions related to the type of rare disease, the type of genetic condition, or the type of molecules that we're specific interested in. So it's more about seeing where the field evolves and then doing some incremental changes so that it become more useful and more helpful for the specific application we're interested in.
Great, thank you. Clay?
I don't know. I feel like I have to change the question because I've kind of been in AI all along.
Yeah.
In the sense that it was my passion in high school. In Italy, to graduate and to go to university, you have to do this final year big test. It's a nightmare, and you have to do these big tests. So I put it. As part of it, you have to write a thesis or present a thesis, and my thesis was on AI, and it was connecting history of science, philosophy. I went to a humanities high school, so I had to do all of that. And it was on AI, so we talk about Penrose and whatever. And then in undergrad, I did a lot more, I shifted my interest to the connected field of information theory and then back to AI. So in some sense, the question for me is, how did I get into biology? And how did I stop doing biology and then got back to doing biology? So as I was deciding what to do for my PhD, basically my advisor basically said, you can pick like pure AI stuff, or you can pick like some CompBio stuff. And the pure AI stuff sounded very exciting, but the CompBio stuff felt like it was going to change somebody's life potentially, because my PhD was in a translational neuroscience institute. It was like in an ALS institute dedicated to the study of ALS. And so we met, the families of patients all the time, like and donors, and it felt very important to work on that. So that's how I worked on that. Then I worked on it at MSR for a bunch of years, and then it felt that there was this massive gap and widening gap between the complexity of stuff people were working on in AI and machine learning at the time and what people were doing in computational biology at the time. So it felt like the methods were just fixed for a while. And particularly in statistical genetics, like there was, every flavor of linear mixed model. And at some point I got bored. And so I started working on other things, specifically automated machine learning at Microsoft. We launched a product. It was a big thing. And then a few years ago, I noticed that the gap was kind of shrinking again. And people started working on really exciting things in computational biology. And so that's how I got back. As to how I stay updated, much to the displeasure of my team and our cluster administrators, I do spend at least half of my time coding and actually doing like work. Like not that everything else is not work, but doing practical work. Sometimes the best way to understand the capabilities of the models, the delta between what people claim on Twitter or X, whatever, and the reality is actually use the thing, do the thing, train the model, see what it does, and that's the only way for me to really grasp.
Yeah, I think, I don't know if it's because I'm getting older or the technical work is becoming more difficult, but I see there's like a widening gap of like, managing an organization and understanding the technical work. And I don't know.
Although, thank goodness for the generative AI tools that let me code, because basically every now and then some package fails installing and I throw it into O1 Pro and I say, just figure it out for me. Like how do I pip install this?
Yeah, I don't know if it's rotting our brains or helping us go faster, but
one or the other.
One or the other. All right, Jesse, same question for you.
Yeah, thanks, Andrew. Yeah, I think so. I came to AI through an interest in national security, right? That's sort of my background, my focus. Where I'm primarily interested are the potential existing or past security implications of emerging technologies. So I started, I did this big project that with collaborators from Stanford on the biosecurity implications of genome editing. This is right when CRISPR was hot, and we were looking at other tools and techniques. And that consumed my life for maybe like 2 or 3 years, and I had to learn everything about genome editing and the security implications, right? And it became increasingly clear to me that that there was this, as many of the other panelists have spoke about, this kind of convergence of AI and bio, right? So increasingly, if I needed to know something about bio, I needed to know a lot more about AI. And slowly, it was kind of like maybe mission creep, where the more I started to learn about AI, it became unsustainable for me to be focusing really on the bio, right? Like it's hard to do both, to be perfectly honest. And there are only so many fire hoses one can drink from, and like this space is and so now we start to see that. Like even if one is focusing only on, or not only on, but primarily on AI and bio, that like the convergence with AI or AI plus, I think this is what AI plus Y or AI plus X or AI plus whatever is like 10 fire hoses anyways. So so I still have been thinking about and working on the security implications of artificial intelligence. But I've been doing that, and so what got me to really what the hook was, right, was that that the U.S. Department of Defense several years ago released these responsible AI principles, these 5 principles, and the then Secretary of Defense said, the Department of Defense does AI, but we only do responsible AI. I thought, oh, that's interesting. What do they mean by responsible AI, right? I knew the people, I knew the woman who led the team that wrote the principles and so on and so forth. So I got to, as I mentioned in my comments, I got to thinking about measurements and metrics of responsible AI. So if the Department of Defense is only doing responsible AI and the AI needs to be secure, safe, trustworthy, governable, and equitable, those are the 5 principles. How do you measure that? So how do I measure governability of an artificial intelligence system or a model, whatever, application? And that got into some really interesting technical questions, right? So and that technical work continues. I mean, right, there are still folks that are trying to work out this problem. So as I was thinking about kind of AI metrology with respect to responsible AI, it got me thinking about it in respect to this space. So how do I stay current? I think it's really helpful being in a university environment. And I would say that who has the kind of the most voracious appetite when it comes to knowing all the latest and greatest research when it comes to AI, and it's often students, graduate students, but undergraduate students as well. So I stay, to great credit to my students, that's how I stay up and current on artificial intelligence.
Great. Thanks, everybody, for sharing that. So I think we now will have a break for 10 minutes? 14. For 14 minutes. So please enjoy your 14-minute break. Thank you to the panelists.
We'll come back. I guess I don't really see anything because I see Andrew's coming back. Yeah. We have one. No. Yeah, I'll be funny later.
Hi, everybody. Welcome back. So now we're going to move into Q&A. So if you have questions online, go ahead and share them out. And then I have a couple questions to start with the panel. So the first question I want to ask is the last, few weeks or few months, depending on your field, have had a lot of changes. And just to list out some of them for everybody in the audience, I think the reasoning models that came from DeepSeek kind of have a pretty big impact because it's both an open source frontier model that's competitive with the best proprietary frontier models, and also the algorithms in the paper have really translated to open source work on language models, and we're seeing very large gains in sort of a new category of language models. There was some good work, I think, on things like there's the co-scientist paper from Google, which came out yesterday. And also, I think just the pace of the field was a little bit slow early in 2024, which, I don't know, depends. If you're coming from biology, it's always fast. But I think we've had a lot of developments in the last couple months in AI. So first question for the panel is, what has been exciting for you, or what do you think has had the most impact on your work in the last few months in AI? So start with Jesse here.
Yeah. Okay. Great. Thanks. Yeah. So the DeepSeek, I'm still trying to. So okay, DeepSeek's been exciting to me, and here's why: is that so I'm sort of still so I think we sort of still don't know the whole story, but meaning like, did it really was it really as cheap to train as is suggested? Did the firm leverage previous resources? How are all of the chips that they bought? But I think what's really interesting to me is that it opened up a conversation. It was, I think, let me step back. I think it was until DeepSeek, we can think about it. It was until DeepSeek taken as not a given, but a seldom challenged assumption that the model is that you need to spend a ton to train. The models were huge. The proprietary models, the closed models, were winning the day. That and that the U.S. really needed the infrastructure, right? That is like, shovel-ready infrastructure in terms of data centers, power, so on and so forth, and that was where major investments were going to go, right? We can think about, the announcement of Stargate, for example. What, 500 billion was theoretically committed to that, and 100 billion in hand was what was claimed. And DeepSeek, if there's one version of the story where that narrative was challenged, where it was a smaller model, maybe used distillation and kind of leveraged other models, and OpenAI's GPT in particular. Right, but my point being here is that I think it's really exciting that it opened up the conversation and challenged our assumptions about what is going to be needed to make the breakthroughs that occur in the future, right? And that's important not to just AI, but as we think about using AI and the cost of developing AI for things such as drug discovery, right, or biology. So that to me was really exciting. Again, I say that with a kind of big asterisk or a caveat, meaning being that we sort of don't actually really know that much about how DeepSeek was developed, the price of it, the size, all these things. But I think what it did do, and what we do know for certain, is that opened up the conversation to allow us to have a broader aperture for thinking about how we develop models going forward in the future.
Same question.
Well, first of all, thank you, Jesse, for taking the first stab at it, because you asked a good question when the thinking models take a long time to think. So when O1 gives me an answer in 3 seconds, I feel like I wasted its time. But so thank you for the time. So I think on that note, test time compute. I think that was the big thing. Historical perspective on test time compute is when a lot of the field used to work on computer vision and vision models, test time compute used to be like, oh, we augment the data, we throw it in, we take some kind of consensus out or ensemble out. And now test time compute actually is an incredible orthogonal component of scaling. It's semi-orthogonal. You need to have a good base model, but like if you can spend the time on test compute, you're going to get so much more performance. And it's interesting in the context of life sciences, biology, biosciences in general, because we are working on problems that are worth the dollars, right? So for a lot of like commercial use cases, you don't want your model to think for 5 minutes to rewrite your email. Like you may as well just do it yourself. But when you have like an asset, like a drug asset worth tens of millions of dollars, like up to that stage, if you have to spend $200,000 in test time compute to just think thoroughly about like all of the evidence and put it together, that's going to be like really, really life-changing. And so for us, super exciting because now we can spend time thinking about things worth spending time thinking about.
I think the test time compute is so fascinating. So I live in San Francisco, and there's a lot of like rumors about what's coming up in AI being there, and people are talking about test time compute on the weeks scale, which is crazy to me that you can like have an LLM spend a week thinking about the response. So I think it is exactly applicable in life sciences. So it's a really great point. Thank you. And James, same question for you.
I want to draw 2 observations you have. One is the open source nature of DeepSeek. It definitely has generated a lot of excitement when it's like get on the news and when some of the paper and algorithm are being discussed. We're just seeing a lot of our scientists and other scientists we work, they got excited about just trying it out and exploring it. And there's like various other tools being developed, of course, in parallel. That actually I see one challenge here is there hasn't been an easy way to adapt it to the general flows of drug discovery, right? Like you can ask them a literature research question, you can ask them about like some observations, those they can help you. But in terms of, hey, like DeepSeek or whatever, or ChatGPT, how like this molecule is not specific or potent enough, like how can we get it better? The answer doesn't help. But it does, I do think a lot of these models and they have access to the data and they have the access to the potential that it could work. The engineering to get it adapted, it still presents a high barrier, and there's a limited set of talent who can use it, right? While like there's many millions of users and everybody being play around with it, but very few people have been able to adapt it to the specific use in terms of designing a better molecule, advance it faster. So how do we bridge the gaps there? And what are the tools that are required to make it easy for particularly people like myself who kind of are late in the game compared to many of our speakers? Like how do we catch up more easily? And what are the resources and tools to enable a broader and more convenient access here?
Takes a second.
I agree with what's been said, so I'll just add 2 things. One, building on what you said, this doesn't relate to a particular, paper or moment, but I feel like there's a sort of agentic AI movement afoot right now, and we're certainly sniffing around there. So shifting from us telling the platform, okay, here's what we'd like to work on, here's our target compound profile, what are your suggestions, to here's the general area we want to work on. What do you think we should work on? So that's been a really interesting shift. We're very much at the beginning of that. but that's one piece in terms of what's going on currently that's impacting us. I think the other relates to what you were saying before, Nicola, about this shift from it just has to be more and more data to what data. For us, that happened a little farther back in time in AI terms, not that far back in time with AlphaFold, where we looked at what AlphaFold predictions said relative to what data we were gathering from our own cryo-EMs and seeing where it was strong, where the gaps are. And so we're now directing our cryo-EM research to try and fill in the gaps rather than just sort of blanket the whole space with data.
Yeah, that's great. All right, thank you. And now I have a question for you, Alex, actually. So at Generate, you guys have built this Chroma model, which has been really exciting, and it was a really cool model, and then you guys followed up with great validation. But I have a question about the drug discovery process at a company like Generate that is sort of AI first. What do you do differently, or do you do anything differently after you've developed like this hit biologic binder in the later stages of the drug discovery process? Do you still use it? Is there any specific AI innovations you try to use later on?
It's such a great question. So let me start with the Chroma piece and then with the what happens later piece. So for those who aren't familiar with it, Chroma is one part of our overall stack of models that deals specifically with generative AI and sort of specializes in de novo, so proteins that have never existed before. In our pipeline more broadly, our first molecules are all in our optimization stack. So starting with something that already exists, defining what our target compound profile is, and then using the optimization stack for multi-parameter optimization. So for example, for our COVID molecule, we said we want neutralization of these variants. We want these drug-like attributes in terms of, viscosity and self-association and those sorts of things, and optimize using that stack. In the de novo stack, we start with a question where there's nothing, no starting point at all. The platform suggests a starting point using Chroma and other programs that we use with it, and then it ultimately shifts into optimization. So in both cases, it's a funnel that leads to the same techniques. Those use both wet lab and dry lab data, as well as, when we don't have it, structure. And so we're doing wet lab experiments in a learning loop. The number of variants we test varies very broadly. So for a sort of easier problem, we may just be in the hundreds, less than 500. Whereas for de novo, we'll do 100,000 variants so that we have enough hits to then optimize on something. So usually we do various numbers of cycles. So if it's an easy problem, it might just be 2. If it's a hard problem, it could be 5 or more. That then gets us to ultimately hundreds of variants. We narrow those down to about 10. And then from the 10 to 3 to 1, that follows more of a conventional drug development process. So I would say the input from AI decreases in those last steps. Basically, when we're moving into the selection of our lead into IND-enabling experiments, then we go into the sort of deeper science, more low throughput, using mice where applicable, those sorts of things. Did I address what you were asking?
Yeah, that's exactly right. I mean, I think this is just an observation for me, but I think the later stages of drug discovery haven't seen as much innovation, and I don't know if people are more risk averse then, or just we haven't made our way up. And I don't know if you can address that in your, or James, you want to comment on that?
Oh, maybe you want to go first?
Sure, I'm happy to. So and I'd love to hear your opinion as well. So I think a big part of it is the closer that you get to the regulated space, the more thorough and frankly more conventional your package needs to be. The agency's job is to protect human health first and foremost. And so the sorts of work that you need to do to dig into safety so far have not taken advantage of these. Now, what I would love to see, and I hear whisperings this may be happening, is for those companies that are working in the safety space to apply what they've learned using AI so that we can apply that in the preclinical space. I think that loop has not closed yet, and the field will benefit tremendously when it eventually does. Things like the connection between in vitro models to what you see in cynos, we need all of that to connect back so that when we're generating our molecules, we actually de-risk those attributes earlier.
Got it.
I totally agree with what Alex is saying. The challenge is almost like one, you're getting ready to the human, whether it's a regulatory or whether you need to convince the PIs to participate, to get people to participate in the trial. That data package hasn't fundamentally shifted. It doesn't matter where the molecule come from. In fact, they actually don't really care where your molecule come from. It could be from your thinking, from AI, from whatever. They do have to pass a set of quite stringent bars. But that said, I do see a lot of AI-driven innovations in later stage of the development. A couple of things we are using and we've seen our industry partner been using is, for example, using AI for clinical trial enrollment. The challenge for a lot of things is like, well, we have a good drug, but by the way, the trial is like taking 5 years because you can't find patients. Trial site is stuck at a variety of different reasons. Now there's a lot of data being collected on these trials, a lot of data being collected on various patients and demographics that you can predict where you can get better enrollment, where the bottleneck could be, and how the competitive landscape evolve, and that can dramatically reduce your enrollment, and in many cases can cut by half, and that's very, very important. Another aspect we've seen is just because not everyone is sequenced at birth. In fact, most people are not. Before that is done, then a lot of diagnoses for genetic disease are based on phenotypical data. It's like some sort of blood chemistry, some sort of imaging data, and you don't know what disease they have. And using some AI algorithms and with patient and physician consent, of course, one may have the ability to recall patients and to proactively reach out to say, hey, like we suggest you should test for this or get a genetic diagnosis or potentially even consider enrolling these trials because of the phenotypic presentation. And that one benefits them, but also accelerates the clinical development. What hasn't been changed, again, is the bar for efficacy hasn't changed, and you just have to hit your primary and secondary endpoints. And AI can help you design, and we just actually, before the start of the session, we were talking about AI can write you 80% of your protocol, but still humans still need to engage in getting the last part done. Another point Alex mentioned that I want to build on a little bit is regarding the safety database. I do think one of our collaboration we've done is with Enlorum in using antisense oligonucleotide to target various genetic targets. And what we're seeing being done there is they have a vast set of safety database and safety experience that they've using a lot of AI algorithm as well. And they can rule out a lot of the molecules that are just non-specific or have animal liabilities. So instead of finding out later in the development or have to sacrifice animals for that, you can rule it out much earlier. The challenge for a lot of, and we definitely want a lot more in the broader field, the challenge is the data availability. Where do you get that, and how do you again integrate into your routine pipeline?
That's great. Thank you.
You want to move on? Another big area, companion diagnostics, that is late stage.
Sure,
I think we're
yeah.
going to see that a lot more.
Yeah. Great. Well, Nicola, I have a question for you, actually.
Them.
So you've been working in AI bio, I guess, for a while now. And one of the things that I'm always curious about, I think actually Microsoft published some early work on this in diagnostics, is what role does explainability or interpretability play, and is it unique in AI bio, or do you think it's similar to the rest of the AI area?
Very good question. My thinking mode will probably have to spin a little bit. My immediate reaction, so I think it's important, and we are doing the kind of mechanistic interpretability of models on our bio models that people are, traditionally doing known in bio models. The problem is that explainability in a lot of the applications in bio kind of reaches a stopping point very much earlier than what you do for other types of models. For instance, in X-ray, models of X-rays, models of pathology, you can see the thing, you can talk to your pathologist, and you can kind of go, okay, yeah, see why it picked up on that abnormal anaphase. Oh, yeah, that's clearly wrong. For other things, such as like things that we do in proteins, I don't know what, it's you can the model, you can tell what the model is latching onto. To figure out exactly why, then you would have to do like a ton of wet lab experiments that we do routinely. But like, that's a challenge. Like explainability is explainability to the human, and the models that now are getting to the point in non-interpretable areas where they know better than we do, kind of like the high-dimensional signals that they're latching onto. And so it's kind of tricky for you. They kind of have to dumb it down for us to.
Yeah. I think it's interesting that protein language models are this like canonical example of I wouldn't know how to start making that explainable or interpretable.
I mean, I was looking at generations from something I'm training, and I'm like, well, that looks like a bunch of letters that starts with an M. It looks right, but who knows?
Hey, for those of you who haven't trained a protein language model, the only thing you know is if it starts putting out polyalanine everywhere, you know you have a bug in your code or it's not trained right. That's about all I know. So Jesse, I mean, you can follow up on that question, but also for my benefit, actually, one of the things I don't understand is what is the difference between like robotics in bio and automation in bio? Because I feel like when I go into like a modern biology lab, there's a lot of automation in like a pharma company or a late stage biotech. But is robots here like humanoid robots? Will that play a role, or is sort of traditional like on the rails automation more important for the field?
Yeah. Yeah, I think. I mean, so there are sort of two ways to tackle this question. One is that, of course, you can have robots that are simply automated, right? And so it may be kind of a slippery distinction. I think that one is that so you have robots that can simply be automated and aren't necessarily autonomous, for example, right? Then you can have robots that have varying levels of autonomy, meaning that they can, do things outside the scope of what we generally consider to be automation, right? That we can, a workflow that's predetermined that it engages in. I think the difference is that is going to be in the hardware. So and this gets to the second part of your question, which is kind of a hardware question in the sense of what are the robots going to look like and what are they going to do, right? And we can have those robots, so they may be humanoid robots. I think that's a really, really long way off, right? That, I guess maybe it's kind of in some ways people think it's like sort of the holy grail of, to have a robot be able to be and act and embody hardware in the way that a human does. There was just I won't name the company, but there was just a big company that did a big launch and had humanoid robots serving as as bartenders, and and they looked amazing, and they were remotely controlled, from behind the curtain. Right? It's a sort of Wizard of Oz. So yeah, I think it'll be that I take your question in two parts, which is that depends on the level of automation, and then if that tends towards autonomy, meaning that robot might that piece of hardware, in this case a robot, be able to act with a degree of freedom and independence that is outside of what simply is programmed into that workflow. And then the other is going to be, I think I'm repeating myself here, is going to be just quite simply what is the hardware going to be, right? So right now it is maybe a hand that can pipette. And I talked about some of those limitations, right? Is that we've probably all seen videos of sort of the ground truth of robots, and they're really kind of limited in what they can do at present. So I think it will be what we'll see is that potentially a convergence in the level of autonomy and the type of hardware that's being used, but I think that it's a long way off that it'll be anything like a humanoid robot. So what at present right now is primarily we think of as on the rails type of automation. Does that answer your question?
Yeah, and it's Thursday morning, so I we'll have to see what announcements were made in robotics today, because I think Figure said that they were going to launch something today as well. So we'll have to double check, in a couple hours. Awesome. So I wanted to open it up for audience questions. Yeah, go ahead, please.
Okay. I want to go back to your question about AI further downstream in the drug discovery and development process before clinical development, which James already talked about. I think there is huge opportunity to accelerate our ability to assess safety before going to human. Because right now on the functional mechanistic side, we rely on preclinical model, and we have to have sidebar conversation about the limitation of those studies. But when it comes to safety, we also limit it, right? We need to use preclinical model, granted, go all the way to primates, but still, they're not humans. But, I hope that there is a huge opportunity for us to get better data from human and really leverage the AI's ability to model behavior of cells, behavior organ system, behavior of actually a digital human in a way that can accelerate our ability to do that tox study before IND. I think that would dramatically accelerate the timeline to get medicine into human, because ultimately we have to test them in human. So I want to hear what your thoughts on that in terms of what kind of data. I have a view on what kind of data we need to be able to model that, and also from a trust and governance perspective.
Made a lot of the key points. In terms of what kind of data we need, as I mentioned before, I think we need to close the loop between the preclinical data, which I'm using broadly, but also, there's several investigators as well as companies developing three-dimensional models, organoids, et cetera, which I'm optimistic about, but we have to look at those data, look at the attendant large animal data, and close that loop, I think, before we can confidently leave out large animals from safety evaluations. I cannot wait for that day to come. So I would love it if, the field could collaborate to do that. I think, to a point made earlier, a lot of these data are within private entities. And so a question remains, how do we either help folks collaborate? How do we get them to publish? Like, how do we actually gather those data? The AI part in some ways is the easy part. It's getting the data all together that I think is one of the major challenges.
Well, I would want to add there, though, I think one of the point I really want to bring out is how do we also get the right data from patients? Not just, obviously it's a new drug, so you wouldn't have human data on that drug, but there will be a lot of data on humans who have been treated with related drug, unrelated drug, in healthy, in disease, or various conditions. I believe those data are necessary for you to really model and predict how a new molecule is going to do. And if nothing else, point us to the right assay to do in large animal, because a lot of what we do really doesn't, I'm not really predicting the toxicity in human, because we wouldn't know, and we don't know where to look right now.
Yeah, adding to the point on the right data and the challenge of a lot of the data being private, the challenge is there's a publication bias. People tend to tell you, oh yeah, this drug is safe, and this drug moved forward, and we did like beautiful 10 experiment. But by the way, there's like a solid another experiment where it was toxic, and things show up, and but you never hear about them. And the saying is, then it's very difficult to use AI or whatever tool to learn about this data, because you're presented a subset. You also don't know what could lead to what. And this is where I think some public entities or other partners could come in, where it's almost to aggregate the data and to make it more available. And then it will really benefit the broader community. It will make the activation barrier for a lot of work to be much lower.
That's great. Yeah, please.
Great. All right. So regarding the data, what are the categories of data that we need to have collective access to in order to really advance the whole field? So this safety data is a great example. So all of these data tend to be highly proprietary. The negative examples are never shared. And so we as a field are failing to advance because of these concerns about sharing. And so beyond sort of efficacy data, where are there other whole categories of data, some omics that I haven't thought of, where if we could come up with a way to share those data somewhat collectively, all of the ships could rise?
I'll give you my thought there. I think there's a whole category of data.
Linda, you'll be on the panel in
I know.
about 45
I'm
minutes, right?
teeing myself up for the next panel's discussion.
We need to add more chairs.
No, I do want you to think about the human data. I mean, the power of AI is be able to handle and analyze complex data. There's no more complex data than being able to look at a human being and a billion human beings and try to extract from that what the biology underneath that is so that it can really inform the R&D process. So to me, that means the tool have to be developed, and you can't train this model without having that kind of data as a starting point. I mean, that's actually more for this panel's discussion. Right?
Yeah, the category data is a little it's a hard question. I think maybe let me just say a few type of data that at least I would be very excited about, right? It's about one is about the real human data, like whatever you can collect on a disease or healthy volunteer, but more a diseased human being, a lot of like what the disease manifestation, what are almost cross-sectional data and longitudinal data so that we really understand the fundamental changes from certain mutations, from certain interventions. Another part of the data is just like the granularity of the data. It's not necessarily about the volume. It's about, for example, getting to the right cell type, and that could be domain specific. The right cell type, the right stage of development, and the right cells. And sometimes getting a small pool that's right is more helpful in getting ones. We've had examples where the therapeutic hypotheses were unfortunately wrong because we pulled bulk data, and the data were generated at the wrong stage. And then by the time you apply the therapy, that kind of target is no longer there because the cell differentiates, the things are gone. And we only find out later on because like the experiment doesn't work. We're trying to figure out why, and we did additional sequencing work. So that kind of data will be very helpful. It's more like not necessarily more, but more categorized, more specific. Yeah.
Great. All right. Thank you. Question here?
I had a question very similar to the one just posed. When you, oh, sorry, is this working now? Okay. I had a similar, almost follow-up, but slightly different, where you were talking at the beginning, a number of the panelists were talking about, we don't always need more data. Sometimes we need better data or better filtered data. And some of this conversation reminded me of some of the work with like minimum information standards and minimum information for various, doing experiments again and making sure everything works. And I was curious, especially from the generative model aspect, what sort of minimum information would you consider necessary for a Gen AI approach to work? And I guess then for more of the chemist side, are there any aspects to that minimum information for generative AI models that are different from the normal, more traditional approach to beginning drug discovery? Thank you.
It's a very good question. When I'm referring to better data, and that's related to the question, I was going to describe, like, there's no particular omics that, like, no vertical that you have to go deeper in or get more on. I was thinking about the horizontal, like, heterogeneity. That's really the thing that our models latch onto. So, like, we're seeing a lot of that with our digital pathology work. We're lucky to work with people who have access to enormous and very heterogeneous sources of data, meaning a lot of patients of different backgrounds, and in digital pathology is very patient-centric, obviously being tissue. But like going down the stack, like at, lower into biology, like at the cell states level, like how do you engineer heterogeneity into transcriptomics? So that's kind of where my comment was going, which is how do you either capture heterogeneity in the world where you have to work with people, or engineer heterogeneity in systems such that, like cell lines, such that you capture all the diversity of responses that you could have to a perturbation or whatever. So that's where I was particularly focused on. And as part of that, the models, like one thing that we're seeing is that you need to inform the model of that variability by providing a lot of side information about what you've done, for instance, to the cell line. But that kind of, I don't know if that answers your minimum information kind of. So I have to be extremely honest. I don't know what you mean by minimum information.
There's just some standards for, I'm remembering there's one for like phylogenetics where you have a specific data set you can sort of say, and then someone can pick that up and replug in and reconfirm what you discovered.
Oh. I'll pass to the next person.
Yeah, please go ahead.
Can I speak to that one? So I think there's a positive example that sort of gets that you answered a question. I don't think the question can be answered generally because I think it depends on the problem being addressed. But Friends of Cancer Research did a really amazing job of pulling together FDA, diagnostics companies, and pharma companies for a certain question that they felt was necessary to the field. And they had a third party aggregate the data. All the parties agreed ahead of time what were the statistical needs to answer the question and what was the minimum viable data set. And they, in so doing, the whole group became comfortable with the data that were shared. They were aggregated and analyzed by a third party. And then, to your point, I think it's Laura maybe, the rising tide did lift all boats because everybody benefited from that question being answered that could not have been answered except by that aggregation. So that is a small example, but at least it's sort of a little ray of hope that if you align people around the question and then you reassure them that, for example, okay, we're going to aggregate these safety data, but efficacy data will not be shared at all. Maybe even the name of or the identity of the drug, only the modality will be shared. Like there are ways that you can de-risk to engender collaboration. At least the glass half full person in me thinks
So was the data shared out like a p-value, or was it something richer than that?
No, it was actual patient level data.
Oh, okay. Oh, interesting.
And that was what was aggregated and analyzed by the third party.
That's
I think
really,
there's- I want to mention the data sphere effort. I think initiated or operate under CEO Roundtable. I think it's called Data Sphere, is actually all the multiple major pharma aggregating their control arm study, from their trials, as an aggregate for analysis. That's another example of sharing data through a neutral third party.
is there concern or is there the possibility of, because that was the question I was going to ask, what level of data is, the granularity is concern that or people would be able to make inferences about maybe not, the actual drug as opposed to just simply the modality based upon the data, or try to develop trends as a result? Fundamental level, how are those concerns addressed to protect proprietary interests, which I think are legitimate in this case and important?
In this particular case, the aggregated safety and efficacy data had already been published in all cases.
Got it. Question from Marty.
Great. So building on that, as a physical experimentalist, it is mind-boggling how fast the AI field can move forward. And then trying to think about this discussion around the need for better data. What are the gaps in terms of actual physical experimental data generation that aren't able currently to keep up with what the AI models would be able to do? Particularly, you say one way to do AI, right, is take a very large data set, historical, and try to mine it for as much knowledge as we can get. But where we've seen things really take off is when you couple active learning loops with on-demand new data generation per request from the algorithm to say, this is what I need to learn as much as I can in the next cycle. So given all the speed and all the exciting advances on the AI front, where are we still lacking in terms of on-demand new physical data generation so that the promise of AI, particularly in drug discovery, could ultimately be realized?
They're just thinking about a lot of the preclinical R&D type. There are two kind of key bottlenecks in terms of where things are just, it has been taking a while. AI is reducing the amount of like total work and iterated loops you need to do, but the length of loops hasn't substantially been reduced. And those are, one is the generation of the molecule. In small molecule, for example, the synthesize, the chemistry is not faster because you're using AI. You likely can get a better chemical route, but if it's overnight reaction, it's overnight reaction. Second is a lot of the in vivo experiment and trying to just validate the PK/PD, validate the safety of that. If it's a 28-day safety experiment, it remains there. And in fact, there's like washout period and all sorts of things. That has not been reduced. And the question is almost like, yes, AI can let you do less synthesis. You don't need as many safety experiments because the ones hopefully will succeed more. But are there ways to even shorten those using some of the tools and maybe integration with robotics and things, either make it faster or make it less costly, and that can accelerate research?
I was going to say, I always struggle with this question because what we take is a little bit of a full stack view, meaning from the machine to the biological system to the perturbation, say, to the readout, there are a lot of things that one can change. And a lot of the work tends to focus, because of just like, human bias, like that's what the lab does, for instance, tends to focus on one layer of the stack. So for instance, a lot of our work these days is on developing better organoid systems. And the issue there has been that there's been this model drift where we have cell cultures or organoids that we grow because they grow fast. And they're not necessarily a good recapitulation of human biology. And so there is this, in reinforcement learning terms or in robotics terms, there is this sim to real drift or gap that is winding, where your simulator, or in this case the organoid, is not as good as a representative of what's happening in the human. And so that's where a lot of our full stack thinking goes, which is let's have the AI optimize the parameters of the actual protocol, including like the parameters you pass to the sequencer and the sequencing depth, whatever. Optimize the biological system, like the organoid, such that you minimize divergence to the patient, and then optimize the analysis of the readout in an active learning loop. But that's where the integration of all of these steps is where we see the big gap.
That's something to that. So I agree with what you said. And to your question, I actually see the Achilles heel in all of this as translatability. Because what we found, at least, is if you want to build an assay and then make it high throughput, with enough effort, you can in many cases. But if you take, at the best end of the example, viral neutralization for many viruses, if you see it in a dish, that actually does translate fairly well into what happens to humans. In oncology, cancer cell death, it's okay from a translatability perspective, depends somewhat on the modality. In immunology, the translatability for model systems, high throughput model systems to what happens in humans is pretty light. So to me, that is the piece where so far AI has not been able to solve the problem. I think it gets back to what you've raised, Linda, about connecting all of this to human data. I see that as a gap I would love to see us work on as a field.
Yes.
Adelina Butchkova of the American Chemical Society. I'm curious on your thoughts on a different facet of this, because we talked about the granularity and the minimum data requirements. But one of the needs we have for both drug design and for safety evaluation is quality data. And when we have data that has a high error bound, if you will, especially for biological outcomes, that's a challenge that's often addressed just by human curation at best. I'm curious whether there are any advances that you've seen that can help us curate data so that it becomes more applicable to that type of model building and helps identify outliers, which sometimes skew models that would otherwise perhaps be able to address some of these endpoints, but cannot be ruled out by any human means that we're comfortable with. Just curious about your thoughts.
Because I always have to remind, well, I always like to preface anything I say in these panels or whatever, saying I discovered my team has worked in exactly 0 ints or 0 drugs. So like I have to be very, very humble because I only go so far. We only go so far in our work. But one of the limitations of a lot of the work out there that we've seen is that people take a very like modality by modality, readout by readout approach to model building. So the model that you build and want to understand biology or the mechanism of action or whatever only sees one slice of reality. And so the two things that I think need to happen, one is increasing modalities per intervention, and the other one is more metadata. And when we say more metadata, people think that they go as covariates in a model, but really like extensive free text information about everything that happened is increasingly going to be the way to start kind of like disentangling all of these various factors. And that's a huge gap. Most of the models I'm aware of cannot ingest any free text information together with the modality that you build.
All right, great. Let's take an online question. If there are any.
Yeah, sorry, you caught me just while I was in between screens. Yeah, it's great how like we're blurring the lines between the next two panels. So one of our online questions is, sorry, they move around. It's about artificial data. Does anyone have an experience with AI to generate synthetic data or digital twins to control for trials? In light of FDA's recent AI guidance across drug development, what have been regulatory acceptance here?
I've only been tangentially involved in digital twins, so I won't comment on that one. I haven't ever submitted any digital twin data to a regulatory agency.
Has anyone submitted digital twin data to a regulatory agency? Okay. What about synthetic data? I mean, does anybody think that's going to play a role in AI and biology or drug discovery?
Yeah, I think the thing is, particularly in the rare and genetic disease space, a key challenge we're facing is just a lack of natural history data and lack of control group. And many times it's just either not practical or unethical to generate large set of control data or long-term control data. So this is where some synthetic data and some predictive models on the trajectory of the control cohort could be helpful. I haven't, yeah, to be honest, there has been a lot of people talking about it. I haven't seen it being actually used in any clinical trial or any data package. So we'd like to see how that could help accelerate things for sure.
Yeah, I think that's right. I think maybe 1 or 2 years ago, I looked at some companies that were trying to sell synthetic control arm design as a service. But I'd be very curious to see, does anybody know? Has that gone through any clinical trials before? No? Okay. All right, a question over here.
Yeah, thank you. So AlphaFold has been very successful in terms of predicting the function of the proteins, but when it comes to the small molecules, it's much bigger chemical diversity on that. Building on the concept of generating better data, So my question is from the panel is there any way we can use AI to achieve the same level of success for the prediction of the molecular behavioral / function of small molecules, either in the physical space or in the biological space? Do I make sense? Thank you.
Yeah, actually, almost to extend that a little bit further, the struggle here is not necessarily what's the structure of the protein look like, or what's the structure or behavior of the molecule. It's when they actually come together, what does it look like? Because the structure can adapt to each other, and there could be allosteric changes as well, and those are difficult to predict, and there's a lot of limitation. I almost feel these models are very good at what they're good at. One, they're rigid structure when there's a lot of structure information. But what we're seeing lacking is almost like a lot of unstructured region and intermediates and transient states where we just don't have enough data, both for just in terms of the protein structure and the protein small molecule interaction. There's just not a good way to capture the fundamental data for it to build a predictive model. And that's where we just been seeing challenges on these model being helpful in getting the guidance.
All right, thank you. Do you have a question over there?
Yes.
Please go ahead.
Can you hear me?
Yeah.
Yeah, I'm Mahin Khatami, retired from NIH NCI, and I have a comment and maybe a concern that giving the wrong data that I called in my publication molecular false flag based on false foundation, we get the repeatedly failed outcome. I published an invention in Federal Register in 2005, and I was comparing the biology of different biomarkers, cancer biomarkers, establishing cancer biomarker database, and it was published in Federal Register. But as soon as I wanted to develop this with a company who wanted to develop the software, the managers at NCI made a lot of different excuses that I would not further that. Going to the molecular tsunami of cancer, you cannot pick one molecule, IL-12, IL-13, IL-1, and then thinking that you are doing something to make a drug for it and to think that that is the answer. The reductionist approaches to cancer is what is failing us. And one hopeful note is that things are changing. I hope we go by the science, the truth in science, not by what has been presented as the science which we are giving to the AI. A false foundation given to AI is going to give you the answer that it's not correct. And you keep finding that we are not progressing in any of the drug discoveries. I'm very hopeful that the Secretary of Health and Human Services would do something to go science-based. My discoveries, the reason I'm a little bit bragging now, my discoveries on inflammatory diseases of the eye, which produce the first evidence for a direct link between inflammation and tumor development is the first evidence. I brought it to NIH, I brought it to NCI, and I proposed that the important role of inflammation in cancer research. And the management says, no, inflammation has nothing to do with cancer. Has nothing to do with cancer. These are all documented in NCI, NIH, scientific document, and also in legal document. And now the whole world is working on inflammation, aspects of inflammation, but we are nowhere to understand actually what inflammation does in cancer, because not we are progressing. So I have a lot to say maybe in the next session. We have to be truthful, and the public expected from us. We haven't given that to the public. Cancer risk index goes up, and we are nowhere to do something positive for the public, except for promoting our jobs, and only one type of job. We have to change the culture of who we know to the culture of what we know. We know very little when it comes to deciding in the decision maker at the top level. We have, there is a lack of diversity in the decision-making roles. There is the science and the public funds, which are huge, is in the hands of only a handful who decide for the rest of the people. Thank you.
All right, thank you. I think there's a lot in there. There's one question from that I'd like to ask the panel is, one of the things that I found in my work is that when we use AI models and we have them draw upon existing data or papers, they're very susceptible or very gullible to information in papers. And in fact, in scientific publishing, I don't know if everyone's aware of this, but there's 250 million papers out there. There's 7 million or so published per year. And so that's an extremely high number of papers. And if you go read these papers, many of them are pretty low quality. And so if you go and you ask an AI system, like, has anybody shown that magnets can cure cancer? There are many papers written on this topic. I don't think any of them are real or correct, but you can actually find this. And this is seeping into these AI models that we're basing this on, I think, some problems in the literature. So I'd be curious if anybody has any thoughts on this, come across this, or ideas about how to remediate this problem of like building on the foundation of scientific literature being a problem.
I was going to ask you this question. So
I actually have a good answer for it, but I'm not on the panel, so I'll let you all answer.
You to be
Yeah,
on
I
the
think
panel for this
yes,
question.
yes, we vote.
So this is a really hard problem because I build systems that do this. And I would say that there's not a short answer, but I will give you a few answers. One of them is that one of the hardest jobs in the world right now is assessing if publishers are predatory and assessing if journals are like acting in good faith. Everyone who's tried to do this job has been run out of their professional careers. But right now, the Finnish publication forum, the Finnish government, puts out a tier list of which journals are peer-reviewed and which ones are high-quality peer-reviewed and which ones are predatory. And so I think that is like a first tier of work on filtering what you feed to your models or what you consider. A second sort of thing is fortunately or unfortunately, a citation count. I know it's a problematic measure and it doesn't always get things correct, but a lot of times citation count is a good signal. And then another really great thing that comes out of language models is they actually can reason about what is a good source. And so sometimes it's as simple as showing a language model where the work came from, maybe what journal it came from, how many times it's been cited, and maybe any corroborating work. But that's just the beginning. So one of the projects I recently did was validating the citation graph of scientific literature. So when somebody cites another paper, is their characterization of the cited work a mischaracterization or a correct characterization. And this signal actually correlates weakly with if a paper is subsequently retracted. So basically, papers that are retracted often have mischaracterization of the citation graph. So there's all these kind of signals. None of them are a direct measure, but of course, the holy grail is to basically reproduce scientific papers in silico or reproduce them as far as you can in the primary analysis. And so I posed a question and answered it myself. I feel like this is bad for a panel moderator, but anyway, and these are things that we think about. And so I don't have a good answer on it, but we've been working on it.
Yeah, it's hard when we've struggled that a lot as well, because we've been trying to leverage these models for target validation, for example. And the thing is, like a 9060 paper that's been around for a little bit, like that may have just laid out the fundamental principles, and that may be right. And even though you have 100 other publications, which sometimes the model pick up, that speak like a variety of other things, those may not be right. Those may not be published in the right journal, as you mentioned as well. I think one practical approach we've been adapting is AI gets you like 80% to generate like 3 hypotheses, and then human gets into kind of curate those hypotheses and test those. But how do we get them better? And what, I mean, also, what is a right paper, a wrong paper? What is a right conclusion or not? It's very hard to judge like on the spot. It's easy when you say their data are not reproducible after a couple of years of work, but if a paper was published yesterday, how do we evaluate the merit of it? That's kind of a challenge.
Yeah, I mean, it could add a sort of thought that's adjacent to this, which is, and I'm surprised that generative AI has come up, but not the proclivity for hallucinations or how to validate a model or trust it. And so one is that the kind of larger problem is just simply how to evaluate the sort of benchmark of a model, how to tell that a model is, and right, so that often there's a kind of a whole industry of doing this, but what we often find with some models is that, right, the benchmarks are created by the individuals who are doing the evaluation of the model and then testing the model and then reporting on it, which I think is like not the best. I mean, in my view, not a really great way. I would never, if I wanted to do an assessment of a student, tell my student to create the exam and give me the exam answers for the exam, grade the exam, and then report it to me honestly. Okay. So we're talking about sort of, we're using, I think, which is interesting, right, is that there are sort of like different models that we think of when we think about models, but I think that's one. So we have lots of different problems here and some potential solutions, whether it's the veracity of the academic literature and if that is reliable, reproducible, if it comes from a vanity or a predatory press, but also on, benchmarking models more generally.
Okay. I mean, I could talk about this for a long time. I love this topic, actually. It's something that comes up all the time. And I'll just mention one thing is that people have found that papers which are like fraudulent papers are published in these paper mill kind of settings. They also will deposit primary data into databases that is also fake. So this is the Cambridge Crystal Structure Database. People found that journals would require chemistry papers to deposit their crystallography data to the CCSD. And so they were like, well, I mean, I need to get this fake paper published, so I need to put fake data in the CCSD to get through it. And so I know that a lot of people say the solution to this is like, don't trust papers, rely on the primary data, but the primary data itself is also containing, is contaminated. So I think it's a really challenging problem. And but anyway, I have a few minutes left in the panel. I want to like abuse my privileges and ask the last question here. So what I would like all of you to answer is, what is the most important unsolved problem in the topics we've discussed today? But I'm going to add some things. You can't say more data. You can't say negative data. Okay, and you can't say we need better targets. Okay, so I'll let you all think for a minute. Okay, so what is the most important unsolved problem right now? And whoever is ready to answer can take it.
Yeah, I actually think it's not necessarily about the data. It's about the human, the talent, and the ability to adapt to the tool. And the challenge is I mean, like, this thing is, there's biologists involved in this, there's chemists involved in this, there's data engineer involved in this. The AI in drug discovery thing becomes a very interdisciplinary area, and very few people are trained with the exact right knowledge. And almost everyone need some levels of retraining and adaptation. And how do we, one is make that available and make the resource accessible so people, when whatever the next new model come from and the next new innovation come from, we can have like bench scientists start to play around it. We can have kind of newcomer to the field to quickly get up to speed. And so far, like adapting these models and tailoring it requires very specific training and sophisticated background. It also requires a lot of fundamental understanding of the drug development, which does take many years. So again, like, is there a way to get it more accessible and more generally available? That to me is the challenge here.
I would add just +1 to that. I mean, this sort of echoes I don't want to sound like a broken record, but it is that how do we create, foster, facilitate, and sustain a competitive, robust research ecosystem in the United States and globally, right? There are ways to do that. I spoke of some key enablers, but I think the hardest problem is actually engaging in creating and training the sort of researchers of the future. You mentioned one way, right, which is kind of a mixture of experts model, so to speak. But there are others, and I spoke about what some of those key enablers are, and I think that I'd just like to foot stomp that. So it is to have a research ecosystem that is robust, predictable, right, free of partisan politics.
Well, because you guys said those ones, I feel like I can say a different one, but I do agree that those are very important. I'll go back to what I said a few minutes ago. Translatability, particularly with respect to the daunting heterogeneity of human pathology. I still remember early in my career as an attending physician, I had a patient with ovarian cancer who had all of her metastases resected. I sequenced them, I stained them, and I was looking at them under the microscope, and I just thought, oh, serious trouble. And that sort of spawned a whole, not just my work, in that period, there was a whole spawning of this field of looking at intra and intra-patient heterogeneity in oncology. It has been less well studied in other indications where biopsying is less routine. But when I look at the mountains that we have to climb to absolve humanity of some of its worst pathologies, if we're going to successfully bridge the gap, we need to solve this translatability problem.
So I'm over constrained now. Your constraints, everybody said things I agree with. I'll go extremely concrete. Like, I do think that the models that we're working with, like the generative models we're working with today, while they're incredibly powerful, the base models are powerful, the test time compute and thinking models are powerful, I think we're still missing one more generation before we can actually, and I don't know that anybody has the answers to what that looks like in terms of the ability to come up with genuinely new ideas. There is a lot of, back to the point of you throw a bunch of garbage literature in, what do you get out? There is a lot of, people call it slop that comes out of these models. Like they just give you a pretty good average response. Sometimes it's decent. One of the real tests for AGI that I think Demis Hassabis at Google, for instance, uses is, given the amount of information Einstein had access to, would one of these models be able to come up with something nearly even close to what Einstein came up with? Like, it doesn't require any wet lab experimentation. It doesn't require any physical world intervention, but yet, like, I think we're still far from that, and that's still an unsolved problem.
Awesome. Well, I think we are up at lunchtime. So let's thank everybody on the panel. Wait, is it lunchtime or did I get that wrong?
No, it's lunchtime.
Awesome. Yeah. Thank you, everybody.
Thanks. So people at Keck will have a no-host lunch 2 floors up in our brand newish cafeteria.
Let's thank
So
everybody on
we'll
the
just all
panel.
reconvene upstairs on the 3rd.
Wait, is it lunchtime? Or did I get that wrong?
No, it's lunchtime.
Awesome. Yeah. Thank you, everybody.
So welcome back after lunch. I'm Nikki Pohl. I'm part of the Chemical Sciences Roundtable, co-chair, as well as one of the panel organizers. I had a great morning session, and we already started bleeding the discussion into this next topic, which is exciting, expanding access, removing the biases from drug discovery. When you think about the things we've heard this morning about lowering the price of or the cost of every one of these portions of drug development, you could start going to the extreme where, for example, instead of having personalized medicine, we'd have individualized medicine, where each one of us is a different disease, which is in reality a true manifestation, and there would be a drug or therapy for each one of us as individuals. So that's when Michael this morning mentioned, how do we treat all of disease as rare diseases as opposed to binning them artificially into categories that oftentimes aggregate people where we don't have a lot of data, and we ignore huge swaths of the US population, for example, let alone the world population. So I got into this for a love of chemistry, actually, and being a graduate student in the 90s when there's a lot of excitement about combinatorial drug discovery and all the automation. And what struck me then is that the hype died, as often happens with these things, but every library looked very similar because we didn't have the chemistry that the robots or the automation platforms could run. And so my lab has been very active over 20 years in trying to develop new chemistries that you could actually use automation on, and making them very reproducible, because that's the other thing that came up this morning. How do we also have reproducible data sets that are trustworthy? And that's part of the scientific incentive system that also came up this morning. And we should give some thought about also when we think about a bright future, what sort of incentive systems do we need to create to enforce the kinds of collaborations we'd like to see that are necessary to drive this field to the next level. We also heard this morning about models latching onto heterogeneity. And that is the other reason why we need much more diverse data sets, and diverse in the thinking about heterogeneous in what way, right? I mean, I would say rural populations in the US are underrepresented in a lot of our data sets, for example, and they're going to have different environmental exposures than somebody living in the middle of a large city with a medical center. So there's many ways that we think about diversity also in thinking about our data sets and the heterogeneity we want, and how do we create things so that we don't have the biases that we have in the very small, tiny data sets that we're starting from in this process. So I'm excited to have 3 great people. One is going to be joining us online to start thinking about these problems of how do we shift the emphasis on how advancements into the advanced tools are used and the data generated from drug discovery process can contribute collectively to also the affordability. Because if everything gets cheaper, that also makes it more feasible to start having drugs for ever smaller population sets. And that benefits all of us, because we are all, even if the phenotype says heart problems, that could be a many, many different reasons for behind at the actual genetic, phenotypic, environmental level, why one might manifest with that particular disease. And I think we have the tools, and we're starting to build the tools to be able to look at this in a much more complex way because of increases in data science and AI. So with that, I'm really looking forward to this afternoon panel session, and I'd like to hand it over to, we'll start with Sanmi from Stanford University. If you can introduce yourself, and then about 15 minutes for your background and some opening remarks on this topic.
Thank you. It's my distinct pleasure to be here today. I made some notes, so I'll just talk through them, and looking forward to the open conversation later on. So in April 2024, a data-driven decision by the U.S. Preventative Service Task Force changed the cancer screening guidelines from requiring or suggesting screening at age 50 down to age 40. And for me, the shift on policy actually ended up having a meaningful personal impact because a few months after the shift happened, a family member went in for their routine screening. And because of these new guidelines, they were diagnosed with breast cancer. Now, while this news is devastating for our family, we're very grateful for early detection. And for me, this is a meaningful anchor between some of the work that happens with data and understanding measurement up to what this means for people and what this means for policy. And knowing how this cancer might have grown undetected for another decade. Because as you know, survival is highly correlated, directly tied to early detection. Now, this is about detection, not about treatment, and we're here to talk about treatment, but we're already benefiting from the work many of you have already done tied to treatment of cancer. And so a lot of this work, of course, academic and meaningful in this way, but nothing helps you think about your work differently when some of the work goes from the conceptual sort of paperwork to real life and does affect you personally. At Stanford, I lead a group we call the Trustworthy Artificial Intelligence Lab, where we think about the foundations of trust and safety in AI and think about applications to science and healthcare. I also serve as president of an organization called Black in AI, which thinks about closing participation and access gaps in the field broadly. And I think that this combination of roles gives me some perspective on both sort of the optimistic part of AI technical capabilities, but also potential impacts on individuals asking questions tied to things like healthcare equity. So I'm not an expert on drug development. I do think I have some expertise on artificial intelligence, but I hope that some of what I'll talk through and the insights we've gained over thinking about AI across lots of different fields may be useful in the conversation today. So the first thing I want to mention, which I think is a little bit counterintuitive, is that I think as we've been discussing today, there's more and more signal that AI could be used to directly help reduce healthcare disparities. And I think it's counterintuitive because the running headline is about AI systems that amplify biases. And I think this is a real thing and is worrying. And part of the discussion is what we can do to make sure this happens less often. However, I think there's some opportunities that I think are somewhat unique to the way AI works that are relevant to drug discovery that can help us close some of these gaps. So the first is that AI, I think, often acts as a mirror on society. So AI systems, when they make biased decisions, are often reflecting patterns in historical data. And in fact, patterns where humans find quite difficult to interrogate directly for themselves, but somehow we're much more comfortable interrogating these patterns when it comes to artificial intelligence systems. I actually think this human bias is something we should harness much more. The fact that we're willing to ask hard questions of technological decision-making systems and look for gaps in the way the systems end up leading to decision-making. In particular, in drug discovery, this often will mean thinking about identifying populations historically underrepresented in things like clinical trials, and having this additional visibility should ideally force us to confront and address these gaps when we find them. A second is the broad idea of systematic improvement. So unlike human decision-making, AI systems by design can be rigorously tested and refined. And so in drug development, I'm optimistic about various ways of auditing algorithms, thinking about diverse genetic backgrounds and diversity broadly across humans. I think we'll talk about this quite a bit more, as I think you prompted earlier. And differences in drug responses across populations. I think this allows us to proactively address disparities, and I think this provides opportunities to address these much, much earlier before, for instance, drugs reach the market. Third is intentional design. So we can program AI systems to account for population differences in things like efficacy and side effects, I think the key here is not about color blindness or blindness to demographic differences, but it's about being intentional about how these differences end up mattering medically. To get there, I think there are a few crucial elements. One is recognizing that technological impacts, and particularly AI's impacts, can vary dramatically by stakeholder. As already discussed, data plays a pivotal role. And as already mentioned, historical biases in medical data collection can amplify existing healthcare disparities. And in drug discovery, of course, there's worries about these leading to bigger gaps across populations. So ensuring clinical trials and genetic databases represent diverse populations are key. Second, and one that I often have to teach my colleagues, I think more and more, is thinking a lot about metrics and measurement. AI practitioners are extremely good at optimizing things once you specify what that thing is. This idea of hill climbing is, I think, actually one of the superpowers of the AI field. Once a measure of success is meaningfully well-defined, the field has this incredible energy for optimizing those things. However, this will go to whatever is defined as a target, whether or not it's the right or wrong target. And so very easily we can optimize for measures that don't reflect, for instance, real-world outcomes, or optimize for efficacy without considering differences across populations, and risking interventions that work well for some people but fail for others. So to make this concrete, I think diverse expertise, diversity in all the different ways, and development teams makes a big difference. I think it reduces the risk of blindness to outcomes across different subpopulations. We need to work quite a bit on evaluation. I think mostly I think about this internal facing to the AI field, but more and more as AI approaches are being applied to drug discovery and other high impact areas. Finding a productive balance between innovation and safety is important. And along these lines, establishing rigorous testing protocols to check for effects across different subpopulations, I think is also quite important. And finally, investing in collecting representative data across different groups. So while we're optimistic here at this workshop, I think building useful AI for drug discovery isn't inevitable. Even on the sort of most optimistic positive side of these things, it requires specific and thoughtful investment in data and in measurement. And choices that we make about these kinds of decisions will determine whether the AI ends up opening or widening healthcare gaps. I think about this when I think about what my family is dealing with cancer diagnosis and our treatment. And I think about how these are not just abstract technical decisions, but really impact lives, families, and communities. And we have an opportunity to ensure that AI-driven drug discovery ends up working for everyone. So I welcome all of us to take this opportunity and think about ways that we can move this needle forward.
Thank you very much. A lot to start thinking about for the discussion. Linda, if you could give us a bit about your background, your thoughts on this topic.
Thank you. It is great to be here today to talk about something that I certainly have been quite passionate about, which is data. You know, not just as a physician and a scientist, now as an entrepreneur, but I actually am firmly a believer that data is critical to, you know, future medicine, developing better and more precise medicine or individualized medicine for patients, and doing so in a productive way. And I think data had a lot to do with it. I am not an AI scientist or engineer. You know, I started out, I'm trained as a dermatologist, actually, starting my career as a private practice clinicians in Manhattan that ended up in academic research, very heavily involved in the Cancer Genome Atlas, which taught me a lot. You know, participating there when I was at Dana-Farber and the Broad, and really appreciating what data, how data can transform an entire field. You know, and how it does so in context of all pre-existing technology. You know, I had a wet lab that really focused on biology and heavily utilizing mouse model. And I spent a lot of my efforts in cancer genome sequencing and understanding how to analyze that data and then how to use that data to develop next generation therapy. And all of that, led me to then, moving to MD Anderson to start, the first department of genomic medicine, thinking about how we bridge the gap between the science of genomic and the practice of medicine when genomic is playing, because they're not equivalent, unlike some of my colleagues who think they're the same. And they're really thinking about what technology can help clinicians on one side, scientists on the other end, converges, so that we can apply that technology. And all of that really led me down to the path of recognizing two things. Data is fundamental. Without the clinical data to annotate any kind of molecular data we generate, we really don't understand what that genomic data is telling us. But getting good clinical data is really hard, and getting right or high quality clinical data is near impossible. And on top of that, in the environment that I grew up in, academic institutions, the kind of data we have is very biased and limited. And in fact, at one point, I looked at just what the TCGA cohort represent compared to the SEER database. It's shocking. It's one of those pie chart with color code that you don't need any statistic to tell you. On one pie chart, you have 5, 6 color, representing different populations. And then on the TCGA pie chart, you have 2 colors. And that's it. And yet, 15 years later, TCGA data is still foundational to many decisions that go into drug discovery and development pipelines. It's better than not having human data, but it is not good enough data to inform such important decisions that have, well, what we heard, broad impact, not just on the success of that drug discovery and development effort, but also the impact on patients. If we don't have that data, we develop drugs, we try to apply that and prescribe that drug to patients that probably should not be, but they have no other option. So you give them the next best thing, what's available. So to get to that individualized medicine that really is equally effective for all the patients, we're going to need a whole lot more data. And so that's sort of one of the reasons that ultimately led me to what I'm doing now, leaving academia, starting Apricity Health, a company at its core, trying to build a platform that connects and eliminate the white space between healthcare delivery, where routine care patients sit, and drug discovery and development, and creating the structure that allow us to engage that routine care population so that we can better understand how they respond or not to a particular drug. So to have something a little bit more concrete, I like to sort of just talk about why human data in this case is really important and the right kind of data. And preface that by saying, I grew up scientifically working on mouse model, and I spent 15 years in sequencing genome. So I'm here to say that when it comes to identifying the right target for us to move forward in productive, translatable drug discovery, we need more than the mouse model, and we need more than sequencing that tumor genome. And think about this in context of drug resistance. We all know about the excitement over immuno-oncology drugs that have delivered cure, that allow us to use that word in the cancer field. But across the board, we are looking at 15, at best 20% durable response rate. That means majority of patients either never responded or developed or failed to respond for a significant amount of time. They develop resistance, in other words. But the way to identify these resistant mechanisms is challenging because we rely on laboratory model system, whether it is an organoid in culture or a mouse model or your immune cell extensively profile or deep omic analysis of their pre-treatment diagnostic sample. That really have not led us to many success. In fact, at least over 1,000 clinical trials over the last 15 years have tried to go beyond the T-cell checkpoints that we have today, the T-cell checkpoint inhibitors we have today. And we have very few, maybe one success to write home about. And that is it's not what we want. So why does that fail? Because we are not asking the right question. We are not starting all these drug discovery and development effort with the right target. And the right target had to come from human. We just think about it. We can engineer a mouse to have an identical human immune system in terms of the DNA. But that mouse is housed in an antigen-free environment, at least they should be. They are not exposed to what every human gets exposed to. How can we simulate the immune system's respond to a drug given to that patient? And if we only have 100 patients that were in the trial that led to approval of the drugs, and we study what the 20%, the 50% that responded or not respond, how can that 100 patient represent the very response that the next 100,000 patient is going to have, living in different environment, eating different diet, which, by the way, we now have microbiome data showing that they affect. They're not just your grandmom's idea that you should eat better. We actually have scientific evidence showing that it changes how a patient responds or how a disease will progress. So I think we have to really allow ourselves, or we have to force ourselves to say, how do we change the structure? Because the healthcare delivery system touches the patients. How do we leverage that connectivity point to generate the right kind of data? And that is not just EHR data. That has to be research consented, sample, and prospectively curated high quality data so that we can understand the response and resistance and toxicity of the general population have to any approved drug. Now, what's the benefit of that? And how's that related to our effort in terms of AI-driven drug discovery? Well, I think that's a lot of data if we can get them. And we're going to benefit a whole lot from the technology of AI to help us make use of that data. Second, I think it will allow the process of the drug discovery effort to have a higher probability of success because you're starting with something that's much more likely to translate to human being because you did not start from a preclinical model system. And then the third, I think by going beyond the typical research population that our system, which is the academic center, tend to engage in, we unlock therapeutic opportunity because there will be resistant mechanism that you don't find if you're looking at a biased subset of populations. By looking broadly and looking at everyone, we're going to find new opportunity to develop therapeutics. And ultimately, if we can do that with every patient, then we have a chance to develop individualized therapy when the drug discovery process can be dramatically accelerated with AI technology in terms of making the molecule faster and cheaper. So I think we need both of those capabilities come together to accelerate drug discovery and development using technology like AI, which come back to a couple of points that I think I want to touch back to what Sammy is saying. I think the heterogeneity of the patient population is a challenge, but also an opportunity. And it really is where technology like AI should shine, but we need to capture the heterogeneity in the data sets. And then the translatability. I totally agree. I love that word that Alex used because we can make we can identify target, we can make molecules and we can cure cancer in the mouse, by the way. But doing so for human is a different challenge. And so we have to think about that, which I believe is what Sammy said, right? The metrics. Is the metric curing mouse, the tumor cancer in the mouse, or is it curing it in humans? And what kind of measurement? Because the poorly, passively aggregated EHR data does not give us the full measure of the biology of the human being. So that translate or reverse translate back to what kind of data do we need? So I think I probably should stop here. I'm rambling. So I'm going to stop here and look forward to a robust discussion.
Thank you so much. I realize one of our biases is also toward human health and not animal health. So if anyone with a pet mouse might actually care. And there's a whole, those of us who've been on agricultural campuses, animal health is also, of course, a huge challenge and all the diversity there. So our final panelist for this afternoon session is Nick Davis, who will be joining us online. And we'd love to hear from you a little bit about your background and then some opening remarks on this topic.
Hi, Nikki. Hi. Can you hear me, by the way?
Yes, we can.
I don't know if you've been following the news, but just to clarify here, don't leave the podium quite yet, Nikki. So I'm actually in the air. I apologize. I am on an emergency travel to Europe. It's sort of the real one, if you will. So I'm a defense investor first. I'm an AI investor second, and I'm a healthcare investor third. So I'm happy to proceed however might be useful, but I think it would be better for us to not just have a blank talking head, but rather to have more of a dialogue, an active dialogue. So I think that was maybe some of the structure that we had discussed. By the way, you look great today, Nikki. So I can see you, you can't see me. We should not lose any connectivity because I have pretty good reception up here. I'll give you a quick introduction. My name is Nick Davis. I'm an investor, but I'm a scientist at heart. I'm interested in solving critical problems, challenges, both known and emerging, that sort of threaten the sustainability of human civilization. Not to be dramatic. AI has a lot of opportunities and potential. Actually, understanding the importance of computational methods and respecting that is what actually activated me and motivated me to pursue my PhD at MIT on the heels of graduating from an undergrad at Stanford. I've always been dedicated to leveraging technology to sort of unlock the unspoken capabilities that are not sufficiently well incented by markets. And markets always get what they incentivize for. We're talking about equity and how to sort of serve more patients, right? With AI enablement in medicine. Unfortunately, you may wonder, why does a PhD in biological engineering who goes to MIT and to Stanford, why does that person become a defense investor? Why not join a VC firm in Boston, for example? Well, it's because our industry is vehemently, terminally, and irrevocably racist. So when we talk about TCG, the cancer cell or genome atlas, right? It's because people don't think about Black or Asian or brown people when they do their research. They don't hire them. They don't staff them. They certainly don't make the medicines for them, but they certainly will use their taxpayer dollars so that they can get reimbursed. So we're facing a pretty critical challenge. And in fact, a lot of the structural challenges, sort of gravity collapse that we are witnessing in the United States is evocative and in fact indicative. It is the side effect of systemic failures in medicine and certainly in technology. I needn't go on a soliloquy about all of the structural challenges that exist in the world today and that have been precedented throughout the past 25 years. Maybe it would be best to turn our attention to more of a group discussion about some of the solutions. I will say here, though, I caught just the tail end or maybe half of Linda's time. I am much less optimistic than Linda about the utility of AI enablement, but not because her ideas lie in the wrong direction. We are absolutely directionally aligned as scientists, right? One of the things I've had to disabuse myself of is scientific thinking when thinking about markets in the United States. So rule number one, markets always get what they incentivize for. Rule number two, and this is one that I always think about. This is actually not something I came up with, but something that my mentor taught me. Markets can hallucinate for longer than people can stay alive and for longer than you can stay solvent, liquid. And I always think about that because we've been talking about diversity in clinical trials for what, 30, 40 years? We've been talking about the importance of, I don't know, maybe thinking about 51% of the, doing research that addresses 51% of the population, women, by the way, in case anyone has any questions. But that, for I don't know how long we've been lobbying for that. These changes don't happen. So when I'm thinking about solutions for these systemic problems, the solutions that most come to mind, they involve force and arbitrage, to sort of borrow language from my defense contracting buddies, who are not having a great week at the Pentagon this week, I might add. Arbitrage, by thinking about the reality that we've inherited and thinking about what that reality actually responds to. And one of the things that I've thought about, and one of the reasons I'm on a plane right now, is that it seems that our current White House is obsessed with money, not with productivity, not with efficiency, not with normalcy, stability, safety, truth, accuracy, but money. And so if we have any ideas about how to better use, better train, better enable AI innovation, it has to involve shitloads of money. I divest my time.
Thank you, Nick, for that perspective too. So now we're going to the panel, and I guess I would recommend that we start with the technical challenges, and then we will go to the more meaty problem of the human challenges and the incentive systems, for example, some of the things that have come up this morning and just came up in Nick's comments also. So Sammy, when you think about the vision that you have, what kind of technical challenges do you see as the real meat of what we can't do it today? For what reasons? What are the technical problems and hurdles that we would need to overcome as a field in order to get there?
Yeah, thanks for that question. So a lot of my current thinking of this ends up anchoring a lot on the measurement side of the problem. So at least in terms of once the problem is reasonably, to Nick's point, resourced accordingly. The resourcing part is a big deal, by the way, as hopefully we'll talk to a bit. And sort of problems are framed as we've been discussing. For me, the biggest gap I often see is the measurement gap, which is again optimizing like misspecified or incorrect problems. Because again, the incentive structures on the technology side often don't align all that well with focusing on what matters the most in real world terms. They just focus mostly on sort of optimizing, hill climbing on some defined target. And so for me, I think often what I find most valuable to think through and help frame problems is, again, this sort of measurement thing. Another one that I'm thinking of Nicolo's conversation earlier today, this question of where expertise is most useful if you're going to put AI within sort of these broader systems. And I think for many of us in the field, so I think my academic training, I think, is quite related to Nicolo's in that I thought the way forward, and I fought with people on the way forward being, let's get experts deeply inside what the models are that we're building. My sense is that most of that has failed. I'd be curious, actually, as we have up our conversation, what that looks like. Meaning, what we seem to see in the AI ecosystem is that where expertise plays the biggest role and where sort of knowledge of the problem plays the biggest role, seems not to be in sort of the nitty-gritty detail of how we build a technological tool. It seems to be in data curation. It seems to be in the measurement practice. It seems to be sort of like, in addition to specifying like good targets for what it is we're building, and much less so on the internals of what the actual models are. And this, for me, is a meaningful schism because I generally want to think that if we build our models so that they better capture, say, scientific knowledge, we'll do a better job in terms of real world sort of modeling and impact. I think the evidence in that direction is much more mixed than I'd like to admit. And in fact, where we often find the most value bang for the buck and expertise is not in the deep internals, but actually in sort of problem specification, measurements, like the sort of scaffold around the problem, making sure we're tackling the right problems and things like that, as opposed to, can I build a model that matches some very precise, specific notion of, say, binding particular molecules? From my understanding of the AlphaFold folks, this is what they found as well, which I think is also striking, which again was their biggest wins were not trying to put in deep chemistry into the models, but really it was much more on the measurement side. And like the expertise played a bigger role again in the sort of scaffold rather than the internal. So conceptually, I still like the let's have more expertise inside. I think in terms of impact and technical tooling and what matters the most, I'm become more and more convinced that where deep scientific expertise makes the biggest difference is elsewhere. I think that's meaningful for this conversation.
Thank you. Linda, where do you see some of the technical hurdles to getting to this vision?
Technically on the AI side, I think there are way better experts to really comment on that. But I agree with Sammy in terms of measurement and data. I translate what you're saying, or I paraphrase what you're saying into measurement being defining what it is that we're trying to solve, what the problem. And so often we see people trying to use what they have to try to answer a question that really is not the right model, right data, or right approach to answer that, just because that's what they have, or that's what they're good at, or what they're comfortable with. So, coming back to drug discovery, for drug discovery, the measurement we need are going to be, tell me if I'm using that term correctly, it's going to be contextualized data in context. So for me, drug discovery to overcome resistance to a drug, right? So I need to address and understand how a particular individual or individuals may respond or resist a particular drug, and using what mechanism. So that the idea is, if I can define that, then I can develop therapeutic intervention against the emerging mechanism. But that means I need to be able to have measurements of something that's happening in the patients when they are taking that drugs, and knowing that ultimately whether they responded or not to the drugs. And so now that means, it isn't just the kind of data. In order for me to make those measurements, it's not just the routine care that they get, because in a typical practice of medicine, you are managing the patient on the drug, but you may be getting their blood sample. You should be getting a blood sample to monitor toxicity, all the standard of care, but you're not looking at that sample or rebiopsy, looking for other mechanism that may be emerging, either during or at the end when they develop, no longer responding to their drugs, right? So you're not measuring the things that really matter to me if I am there to develop drugs. And so EHR data, not useful at all from a drug discovery perspective. I mean, not at all. They are not the primary thing that will lead me to, this is a target I'm going to identify. So, but we are tracking these patients. We are taking sample from them, but we're measuring things that are, for practice of medicine, important for their patients, but they're not, well, we're not measuring things that are actually important from a drug discovery perspective. So what I've tried to do in the University is developing that real world research protocol so that we have a chance to engage the patient and say, well, in context of you getting your routine care, you're already getting blood tests every time because you have to before you get your next dose of treatment. Or if we just get an extra tube of blood, okay? Not even an extra stick, it's extra tube of blood. But now I have research consent on the extra tube. I mean, there are residual blood sample in every clinical lab, but they are not consented for you to do anything other than their clinical care related assay. So you need a research consent that allows you to say, now I have sample from this patient that I can generate research data. What's that? Single cell sequencing, proteomic, whatever omic that you want to come up with. We have technology now that allow us to now generate the kind of data that actually can help me answer my questions. And so that's sort of, I think I'm interpreting that framework, right? We can just aggregate routine care data and try to say that that will help us accelerate drug discovery and development because we need a different kind of data. And we need data that's longitudinal if I'm looking for resistant, because resistant by definition isn't there before you treat them with the drugs. And, you know, so the pre-treatment sample is not going to tell me how they're going to resist that drug. And then longitudinal sample means that it's not one time point because I don't know when that resistance is going to emerge. I mean, I think statistically it's like a complex system question, right? Mathematically, when does the tide turn between the cancer versus the host in fighting the cancer? So I'm going to have the ability to have multiple time points. So what are the measurement that I need to take? What technology allow me to get the kind of measurement that actually makes sense? So to me, we went backward in saying, now we need to build the platform and capability that allow us to get just that kind of data, not whatever data is aggregated out there, but specifically and do so prospectively and have a way of ensuring all the other quality measure along the way. So that's sort of one specific example of how I think I'm applying, you know, the framework Sammy talked about to, you know, get the right kind of data for the specific problem that we want to solve.
It sounds like you're also arguing then for lower cost diagnostics and lower, easier, cheaper ways of getting all these data points.
Yes, I think, you know, we were talking at lunch about the difference about being an academic or being an operator, right? You know, if I were doing this as a research study, and many of my colleagues and friends have, and I've been there, I get sample, valuable sample from a patient, or I get a consent from a patient, I'm going to want to do everything possible on that patient, everything. But when you are taking a different perspective and want to think about diversity in terms and heterogeneity, which means I don't want just, you know, sample from this 10 patient, I want 100. And you're talking about patients that may have a very different activation level to participate. So how much of burden can you ask them to take on so that they will voluntarily consent to research? When you put those things together, you have to design a very different research protocol. I call them patient-centric research versus researcher-centric research.
A very nice way of putting it. Nick, online, what do you have to what that you see as an engineer as the technical challenges? We'll talk about the incentive structures and systematic problems as a second part. But for now, what do you see as the technical challenges that are keeping us from this unbiased drug discovery?
Well, the technical challenges actually dovetail quite nicely with the incentive structures. I think Sanmi pointed this out. You know, if I talk to if I talk to any of my folks at Anthropic or OpenAI about the actual utility of their products and the product design, especially any of the folks on the, you know, members of the technical staff, as they would say, it leaves a lot to be desired. Most people don't really care about the inference methodology or the training set that folks have structured for their super large largest all-time high multi-parameter all-time token score for their Evo2 model. People want to know, how the hell do I use this thing in research? And I just keep seeing more and more funding, and I feel like we are on a race to the bottom. I think we are on a race to a bubble. And it's kind of, it's incredible to watch, by the way. I'm kind of watching this semi as an outsider, outside of, other than just sort of placing bets, because America is the world's best casino. I'm looking at, obviously, an academic bubble, right? So we're looking at the funding crash. And some of that, by the way, is some of this sort of scrutiny about the academy as an institution, right? Stems from the fact that modernizing academic research is actually really, really hard, mostly because you just don't have the capital. So it's actually a self-fulfilling prophecy as institutions continue to come under pressure. And now I'm seeing news that, for example, at MIT, all non-essential hiring frozen. If you look across the nation, graduate students are having their admissions sort of put on ice. Not great, not great to not have new talent coming in to be able to address some of these technical problems, by the way, in terms of agility in deploying new methods, right? But on the other side, we have these sort of techno wonks, right? That are privately funded, and they have, for example, in some of these private research institutes, we just sort of see folks turning the crank. And I'm thinking, is that because people just lack other ideas? They just have to train, train, train. Is it because people want to be the first or the best? They want to have the biggest models? The thing that I'm missing is the meta science understanding of like why people actually go into science. For example, I don't care about having I don't even know what the most state-of-the-art mass spec would be today, but I don't need like a Lumos, time of flight, blah, 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 mass spec. And I don't need like an entire room of them, right? An army of those mass specs. I really just want to be able to measure my sample and have, some pretty good assurance that I'm quantitatively accurate and have good enough depth of coverage of my sample, and be able to tether those measurements with other measurements, other multimodal measurements that may be relevant for clinical discovery, right? None of that sort of tethering or meta science, product design, really seems to be happening. And when I discuss this, by the way, with the federal government, which I've been doing quite a lot, people kind of look at me with like kind of blank stares in their eyes. And all they do is sort of reshare the same articles on LinkedIn and on Twitter, or X, sorry. I don't want to get thrown into the chamber, on X, that everyone else does. So I'm just sort of baffled about where we're actually going here, because the usability, the utility, as Sanmi noted, is not quite there yet. Maybe people think they'll win a Nobel Prize prematurely. Maybe that's why they're doing it.
Nick got back into the human side of things, which is hard to talk about science without talking about human beings, because that's the whole goal of this. And Sammy, you gave a very nice example to start all this, the patient who's ill now, and that we want to be able to help in a way that can be as humane as possible. So where do you, when you think about the technical challenges, now start thinking about the human challenges, both on the patient side, on the doctor side, on the researcher side, funding, however you want to interpret that. Where do you see the challenges there? And I should say to the audience that after this, there's going to be a future generation, a workshop for those of us in the room thinking about, okay, what are these scenarios? So we need to think about in the best case scenario, the worst case scenario, where can this AI and drug discovery lead us? So you have the floor now, Sammy. Where do you think the challenges are? Obviously, you can't say them all, but from your perspective, where do you see some of the biggest challenges in getting us to where we want to go?
Yeah, thanks for the question. A nice hard one, but I think we've engaged a bit in at least my broad view of what is often hardest. As mentioned, as discussed, science is fundamentally a human exercise. AI in particular, as part of the scientific enterprise, for sure is. And so all of the biases and all of the incentives that affect how we think about both what's important, where we put energy into, all that stuff sort of immediately has an effect on focus. Maybe a version of what I think we've been discussing a little bit today colloquially is sometimes called sort of Goodhart law effect, where there's some true good outcome we were trying to get to. There is some proxy and everyone just sort of overloads in that proxy. I think in the AI field, the best version that I can think of is, again, this idea of just size. So the main target not being impact on a real problem, impact on, say, drug discovery, but the actual headline being I trained the biggest model with the most number of parameters, and that being the thing everyone sort of engineers and works towards. And that gap seems to me a norm gap on what we value and how we think about how to shape incentives towards values that have clear connection to impact in the real world. And those are hard to shift. I think those come up over time with institutions and what ends up being recognized and what ends up being sort of praised and what people get prizes for and the sort of little trinkets we get along the way in our journey. And shifting those is tricky. I do think institutions play a big role or can play a big role here. So think about what they incentivize. I'm sure we'll talk about funding and resourcing. So what ends up being funded and sort of how that funding is evaluated in terms of impact. So are those impacts, again, narrowly scoped in a way that maybe makes sense from a technical lens, but is too far from either being meaningfully connected to either scientific impact or human impact in the end. And yeah, these are hard problems. I think many of us are thinking about how to engage meaningfully with this. To this point also, again, who we end up thinking about impacting. So as we talked about at the beginning, and I think as Nick very eloquently said, most of the ways we think about building science for medicine and drug discovery, again, is often myopically focused on just small segments of the population. Part of it is data, I think, as you've already talked about, Linda. And also, part of it also is, again, I think, just the targets, what we think of as important, how we think about measuring success. And so, again, to me, these are norm issues. These are sort of ecosystem issues. Are the solutions top down? Are the solutions bottom up? I think I'm open for a robust discussion and maybe some ideas on solutions down that road.
Linda, where do you see not the technical challenges, but the human challenges for AI and drug discovery to be less biased?
I agree with what Sammy said, I think, and then what Nick was saying, because the human component is the hardest one. I think the technology today allow us to measure all the things that we need to measure, or at least we know how to use in terms of translating that into drugs, but getting the humans to play the part and then, so I think there are the investor, and I'm going to let Nick talk about that. There are the providers and the patients, right? And I'm putting aside the drug developers because I think the drug developer would love to have the data, would love to have the target, would love to run, apply their capability to it. So how do we get the providers / even the researcher to study, let's say, the less biased population or less studied populations? And so the incentive system is at play because you need to publish at the top tier journal, because if you don't publish, you don't get your promotion, you don't get your grant. And what kind of research do you do? Is it answering a critical questions, or is it doing the novel thing? Because you don't get into top tier journal if you don't have novelty. And to me, novelty and validity is inversely correlated. I still believe a target is less likely to be, how's it go? The validity of a target is inversely correlated to the time since its discovery. So there's more time, more study done on it, the more likely someone would find something that counter the conclusion on that target. And I don't think that means science is wrong. That simply means there's heterogeneity in biology. And we all know that's to be the case. So, but if your incentive is to publish, and in order to publish a top tier journal, it has to be the first, had to be the novel thing. And with novel mechanism, as if there's infinite mechanism in nature. So maybe you make up some along the way. No, I don't mean that. I'm sure that doesn't happen. And you chase after that. So you use what's available and you're responding to the incentive system. Even though if you take that as way, and Nick said, if you put the right incentive system, they may be very interested in studying a population that is not what everybody studies. Studying a known mechanism and how it can be generalized to a less studied population. But those things don't get you that novelty and that high profile publications. So that's on the researcher and the provider level. I think on the patient level is, why am I contributing to research? I think we need to rethink the premises of research consent, because we, as a system, for good reason, said that consent should not be induced. In other words, it's voluntary is blocking on the word. It is something that you contribute not for your own benefit, is for future generation of benefit. I think that narrow our patient population down in terms of the subset that's altruistic. They're altruistic either because they are at that point, their circumstances make them feel like that's what they can do for the next generations, or they're inherently altruistic and willing to help science in general. But I think a big proportion of our population or our society asks, why would I be doing that? Because now I have to go to a doctor 10 times more. I have to undergo this procedure and that procedure 5 more times. And I'm going to lose time from going to work, and so have to, take me to doctors 5 more times and have, days off work. Why am I doing that? So we don't give our patient an incentive to participate either in clinical trial or in foundational translational research. So I think that's a problem.
Already started alluding to some of the human problems, but where do you see, you could just summarize it in the top 2 or 3 incentive structure systems. What would you want to see changed?
Well, I don't know if anyone's been following the news, but science is being deprioritized by regulators. And in fact, I've shared this thought privately with some of the panelists the past couple of weeks. It's not the responsibility of private investors or privately funded organizations to ensure that public goods are available, right? It's also not the responsibility of private institutions to ensure that the public actually cares about what they're doing. But, big but there, if private institutions sufficiently drift away from their contracts with society, so they deprioritize the importance of communication, for example, and communicating what they're doing clearly and emphatically to the folks who are going to be influenced, right, or who are expected to consume or use the products that they make. So I'm talking in this context of medicine companies and AI companies, AI research labs. If these companies just avoid any of that social contract, then what we get is a deterioration of the fundamentals, because we get a deprioritization by regulators, by government. people should be, this week in particular, people should be protesting in the streets that scientific enterprise in the United States is under attack, so to speak, right? It's being defunded systematically, but they're not. And part of the problem here is because, let's talk about the incentive structures. The people that I know at Anthropic, when I tell them that markets are dysfunctional, they are giddy with glee. They said, I know, right? In the best way possible. I'm not that much of a weirdo where I need to be paid $2 million a year to write code and think that it's a good thing when people are experiencing surges in unemployment. So there's discontinuity between industry, academy. Most people don't know anything about publishing, right? And society. How do you address that? Well, I'm actually hopeful here. I'm going to share something hopeful. I think we are on sufficiently on the onward march towards systemic collapse and deceleration of consumption, and I think and degrowth. And I think that this is going to force people, it's going to shock systems out of their comas. And the only solution to move past that will be rebuilding. And I am hopeful that in the rebuilding phase, people will keep in mind the drivers and components of incentive structures that led to the collapse. That is hopeful thinking. It may happen, but the situation of the outcome will be binary, unfortunately. So I don't know, Sanmi, Linda, Nikki, what's your bet? Maybe we'll take a poll here to sort of fit back into America being a casino. I have $100 that hope will reign true, and that in the rebuilding phase, we will be able to align incentives across industry, society, and academy.
So I'd say most scientists, because what we're doing is not going to have a payoff tomorrow, are inherent optimists in many ways. So this would not be a representative sample necessarily.
I know. I'm leading the witnesses here, and I'm preaching to the choir, whichever idiom best fits the situation. Look, there are no point solutions to the systemic challenges. I think one of the things that I've had to digest, and this has been really, really hard, by the way, and trying to be productive and still trying to move the ball into the end zone. I love idioms and metaphors, by the way, is understanding that systemic failures are not personal failures. And so everyone in this room is doing work. We're all trying to advance the status quo. There's nobody in this room who is not familiar with some of the systemic challenges, right? So I try to take a step away from the ascorbic language from time to time to just sort of witness the fact that, there are many of us who are doing the best that we can. But I think that there are things that are outside of our controls. And we may be headed towards a self-corrective or destructive point in the system.
Well, Nick, I want to chime in here. I'm not going to put money down.
Come on, Linda.
Come on. I'll put a drink down. How's that, Nick?
Depends on the drink.
I think the destruction, or what you call it, the systemic collapse, is almost necessary when we talk about some of the entrenched system, in terms of the culture, incentive system. So I agree with you that the silver lining of the systemic collapse is the rebuilding will lead to a better system, whether we are talking about, the promotion and the publication structure in academia, or the approach to incentivize and engage patients in, data sharing or research. I think we need fundamental change in the approach to that. And the only way we'll get there is rebuilding a system from the ground up and having hopefully keeping in mind lessons learned. That last part, I'm less confident. But I hope if we keep in mind the lessons we have learned, with the system, then the rebuilding will lead to something better.
Yeah, I am curious on I think I understand and appreciate the broad premise. I think to Linda's last point, I guess, where do we get the confidence that if things blow up and have to rebuild, people will remember the lessons and build them productively? I'm curious where I guess that confidence comes from. I'm much less confident, I guess. It's unclear to me that history suggests people learn lessons in this way.
AI
Yes.
Maybe
So
the
that-
AI will remember.
Well, we're here, and I guess I hadn't factored into the fact that I may die in the collapse. Thanks for reminding me, Sanmi. But my optimism comes from the fact that look, I am not done. I'm joking.
Memory, not life.
I am not done sort of speaking from the mountaintop. Oh God, not to compare myself to Martin Luther King, but you get the point, right? I am not done sort of really making sure that folks understand that these systemic structures are unsustainable. People don't care what I have to say. What I will say is that some of the heads of the research agencies this week on Monday, they reminded me actually about how little I matter, which is why I think it's actually emphatic that we continue to sort of reiterate and double down on our message. When people tell me during a crisis for the federal government, when people who are employed by the federal government tell you, you're a nobody, and remember that, which as I was told on Monday and this week, it's been a pretty tough week, by the way. It doesn't discourage me from the importance of the message. It actually strengthens and sharpens my conviction that the message needs to be told again and again and again and again. Some of those people no longer are employed by the federal government. Sometimes folks are just so afraid of the inevitability of what you're warning them about or against. Discovery research, by the way, in oncology, to Linda's very well presented fact pattern around TCGA and sort of the structural limitations of the way that we do oncogenic research, and the fact that, well, there is no more discovery or early stage biotech to speak of in as of February 20th, 2025, today. People really resist the truth, and people feel maybe people feel powerless to do anything about it. I think that in this room we have the sort of seedlings of sort of a consensus around the importance of not straying too far off the path of speaking truth to power. Although I will admit sometimes the cost is pretty regrettable, but not more regrettable than systemic collapse.
I'd like to hear from the audience, including the online audience, your thoughts on these questions and any questions for the three panelists also. We'll start with an online question.
Yeah, thanks, Nikki. We have an online question from earlier, and I think Alexandra may have touched on this, and Linda and Sammy also touched on this, but this may just be phrased differently. So let me carefully read this. Can the biomedical research enterprise, including drug discovery and post-market surveillance, go on without reliable patient-centered databases, complete, longitudinal, and up-to-date as the foundation level data? And the question goes on with why. It's like I have to scroll a little bit. Patient databases can hold personal sequencing data, be used interactively like prospective registries with collection from specific patients of data not routinely needed for standard of care, and be linked to tissue banks. So I think everyone's kind of touched on this topic, but this question did come in earlier, so I just wanted to make sure that we got it out there in the open.
Sure. Response?
I'm not sure I understand the question.
How much longer, okay, let's see. How much longer can the biomedical research enterprise, including drug discovery and post-market surveillance, go on without reliable patient-centered databases that are complete, longitudinal, and up-to-date?
How can the industry go on?
Right.
You mean whether there would be no more biotech company if there weren't going to be longitudinal patient data?
Or it might be, perhaps this is the best practice that's being suggested to go for. I think Alexandra touched on this earlier, but I might have misremembered.
Well, I think it's sort in today's market, we see a shift away from investing in early stage discovery because the high risk of failure, and one big contributor to high risk of failure is really lacking the best kind of data that give you the best, most credentialed targets. So I think from that point of view, the industry is suffering, because of the high failure rate and the risk averseness of the investor, wanting something that's higher probability of success, which means later stage. So they want to wait till you get past those hurdles. So for the early discovery company to get past those hurdles, I think Nikki said they have to survive long enough to generate the data and move them to that later stage, or they start out with something that they're more likely to be successful. So I don't think I don't see that. I don't think it's a either or scenario. I think the industry will continue. The question is, can we increase their probability of success? Can we accelerate the time to having a successful medicine on the market? And that's where having better data upfront and better target upfront makes a difference. I can't really speak to whether there will be a company out there that would survive and get funded, even if they didn't have those data and the right targets.
See you. Oh. No, go ahead.
Go ahead.
Go ahead. Thank you very much. Mahin Khatami, as I mentioned, I retired from NCI, NIH, but my discoveries were done at the University of Pennsylvania. Let me begin with a saying of Sohrab Sepehri, who said that we have remained in the dark long enough to speak of light. I enjoyed listening to all the speakers, particularly relating to genetics and genomics and the heterogeneity. And I just have to mention that we have induced cancer in the last 3-4 decades. That was initiation of giving a virus-contaminated polio vaccine in 1955. And despite a tremendous concern of a wonderful microbiologist, Bernice Eddy, whose lab and discoveries were downplayed. She was removed, but she announced her finding in a meeting. Okay, another issue that need to be a little bit thought through is that in 2018, I did publish that cancer is an induced disease of 20th century. We changed the genomic aspect of population. And that is something that we think yes, genomic, environment, and immune neuroplasticity should play a part when we think of experiment. Another very quickly, very quickly, the issue that we are not succeeding actually. 90% of cancer drugs and treatments fail patients. All these years, and after 30 to $50 trillion spent on cancer, and I don't want to go to detail of it. However, the solution to this, which I don't think anybody wants to hear that, because pharmaceutical companies are not in the business to cure people, they are in the business to treat people for the rest of their lives. So there is a big difference. The solution to that is, have we ever, for example, for cancer research, have we ever done a systematic study to show we have done it? I've done it, and I've paid for it, for telling the truth and showing the truth, showing the way to the truth. A systematic approach to show what is the initial phases of changing the dynamic of immune system that goes to changes, developmental phases of immunity, which goes to tumorigenesis, angiogenesis, and then cancer. I don't think I've seen any. We did it in 1980s without being in cancer research. And when I came to Cancer Institute, I was so excited that we did this. And then you paid for it because we don't want to solve the cancer problem. Let's face it.
So yeah, so you bring up a lot of different issues. Linda, since this is more your expertise, do you want to address any specific aspect?
Yeah, sure. I think I'm not going to go to whether the biopharma industry don't want to cure patients. That's a load of questions. But I would say that delivering cure is not and has not been easy. And there has been study of how the immune system contribute, and whether initially or maintaining or delivering durable response in context of cancer development and cancer treatment. Without questions, the immune system plays an important role. And throughout the entire life cycle of cancer, I would say, not in any one specific point, there has been a lot of effort that have gone into understanding and characterizing. Now, there's a limitation to some of these studies because most of the work has been done in model system, in characterizing the cells, immune cell components at different stages of cancer. And, coming back to one of the points I made, that oftentimes when we do that, we don't see the full picture of how the immune system actually operates in the real world human being. But getting to that longitudinal discovery data from, human being that have the disease or being treated in a certain way, and track how the immune system is evolving, whether on the positive or negative side, I think is a missing part. We can only do so much. And there has been a lot done in the field to study that immune response, but in context of less than optimal model system, because the best model is the human being, which come back to how do we create a infrastructure that allow us to have that longitudinal monitoring so that we can begin to understand the immune response, all metabolism, all angiogenesis, all the other hallmarks that actually converges to drive the tumorigenesis process. I think we have really been challenged to deliver a cure in cancer because it's a complex disease with a lot, every process in the body where study, every normal regulatory process can be, and in many cases has been shown to be co-opted by cancer in terms of leveraging that to serve itself at the cost of the host. So I think scientifically and the drug discovery perspective, we need to get that bigger picture to identify not one drug, but combination, because I'm convinced that it will not be a magic bullet. We're going to need to hit not just one process, but multiple processes that are at play. And the relative contribution of these hallmark and cancers are not identical in different patients at different times. So how do we get the data to inform a rational precision combination strategy? To me, is the next frontier in cancer drug discovery. We have been successful in developing molecule to treat a process. And often the increase in survival means that we've been successful in developing molecule that target the dominant process at that phase of development. The unfortunate thing is that cancer can bypass that and evolve to the next phase. So in order to prevent that resistance, we're going to need to have combination, but we need to know of the hundreds of processes that cancer co-opts to drive tumor growth, which ones are dominant or which ones are emerging to be resistant in a particular patient, in a particular time point. And I just think that it's not for lack of wanting to see that survival, overall survival, it's the fact that we are still lacking in our understanding, and that understanding is constrained by the limitation of the data on the system that we're trying to study and cure, which is human being.
Do you bring
Can
up
I
technical
respond?
It is
and
a
human
technical
challenge? Yeah.
Humans. And, you know, so I think is a harder question, a harder challenge than we all thought. Well, the
Can I
field thought
respond?
40 years ago.
So we still have two more people here.
Okay.
Alex, you're specifically addressed in earlier discussion in this panel, so I'll give you a chance to respond, and then James, you're next.
Thank you for that. I was actually going to say here to what Linda said, so I don't need to add more because I agree. I had a question actually about increasing diversity in clinical trials. So if you don't want me to go in that direction now, I won't, but if that's okay, I will. It would seem that it would be to everyone's benefit if we could make help make clinical trials more representative of the populations who will subsequently been treated. As Nick said, we've been talking about this for years. In my own experience as a researcher, I've seen both issues of trust in clinical trials in the context of the legacy of Tuskegee and Henrietta Lacks, et cetera. And then, of course, there are the geographical and health system access challenges. To bring it back to this concept of incentivization, do you have ideas on what we can and should do as a field to solve those or other clinical trial diversity problems that you focus on?
That's a beautiful question. by the way.
Yeah. Oh, so one thing I've already heard from this meeting is much cheaper diagnostics, potentially even things you can do reliably at home. I mean, you think about all the at-home testing that's starting to be there. Imagine if you didn't have to try to hunt, get off of work and hunt down and go to a major city or to where the researcher is. If the test could actually be sent home and that person could reliably do it, and it was cheap enough to do that and robust enough, that would already be part of a solution. Sammy, Nick, and then Linda, do you have any other ideas here along this line that haven't already come out?
I can speak a little bit to this from related work. So thinking about, I do, I guess, scientific and healthcare work on the screening side, and this same question comes up. And one of the ways that I think this point of trust is an important one. I think it does relate to historical, significant issues in the past that I think correctly lead people to distrust large institutions, medical systems, and all that. And I think to Nick's point a little bit, at least parts of it, I think part of the gap is people not understanding what they're being asked to participate in when people come to them, and sort of what their benefits might be, and sort of what the whole point of the whole thing is. In the narrow sense, we find a lot of value in communication. So nurses talking to people and explaining some of the both narrow but also bigger picture of where what the data is being used for, what the point of the experiments is. I think to Nick's broader point, thinking about how to close the gap between public understanding of the scientific work and the work itself is meaningful. And so I guess I heard part of the conversation. I'm actually not sure it's most of what he was saying, but part of it was, again, part of the gap is people don't know what we're doing and why they're giving us money, taxpayer money. And so closing that gap more, I think, makes a big difference. The trust thing in general, I think it's not a solution, but I think it can play a role for people to see themselves reflected in the scientists. So in my AI work, there's something that we think about quite a bit, is that people's lived experiences do matter and how they think about technology. And it also matters in the communication and how people listen. Of course, it's not perfect. People are people and have a whole bunch of diverse opinions and perspectives. But we have found that it can sometimes move the needle in thinking about representation across all the sides of the sort of scientific process itself as one of the ways to close this gap from the scientific and technical work to society more broadly.
Nick, online, do you have a response?
I do, actually. So I have to speak just sort of from my chest, so to speak. It's a saying. So I'm a quarter Black, but because of my skin color, so I'm quarter Black American, right? My mother's half Black American. But because of my skin color, I have had such a phenomenally racialized experience since I was moved from the UK and from Spain as a teenager to the United States. And because of that experience, I sometimes have to be so metacognitive when thinking about science and industry and the potential of industry and science in the United States to do anything in the direction of the benefit to people who look like me, much less people whose actual lived experience is not just bound by their experience of racism, but sort of tied up in the web of racialized systems in the United States. And for most people who look like me, their experience is totally different, right? They're not calling in to the national academies from their jet. You know what I mean? I don't know how to bridge the gap to trust. I really don't. Trust was bad. 10 years ago, or some odd, when I was participating in the first clinical trials that I actually helped design and lead at Mass General Hospital, not exactly like a rural facility. I don't know what trust is going to be like after after this year, for example. I know so many people who are just so devastated and so betrayed by these systems. By the way, I should mention, Albert Bourla, the CEO, my Greek brother from another mother, the CEO of Pfizer, may have heard of it, small little company. He's becoming the head of PhRMA, which is the lobbying group for the pharmaceutical industry. I should mention, most global biotechs and biopharmaceutical companies have left this lobbying group, but he's becoming the new CEO. I like to call them my favorite disaster artist in the industry, in the healthcare industry. He actually said that the benefits far outweigh the costs of the Trump administration, specifically the decisions being made around funding for science and healthcare and clinical trials and drug administration. How do you build trust after having that level of communication with the public? I don't know. I've started to sort of lose interest in pulling at the string of yarn and sort of think a little bit more radically, where whether we like it or not, if we're actually going to have a business that is like the business of medicine, for example, it will have to, it will have to find a way to make and subsidize and commercialize medicines that prevent disease. And we're witnessing, by the way, I think really the first cohort or first generation of medicines that I'd say in earnest and at scale, at population scale, can genuinely be said to be moving in that direction. The GLP-1s, the semaglutide formulations are certainly moving in that direction. We need a lot more creative thought. And unfortunately, we need a lot more government participation and attention and commitment to solving some of these problems. I would say, Alex, your question actually dovetails quite beautifully with the question from the audience, the first question. The industry, in terms of treating disease, is just not very good at dealing with not only diversity of patients, but the diversity of thoughts and the diversity of findings. So if we want to unspool ourselves from this web that feels unconquerable, we may actually have to move in the direction of prevention. And that is such a radical thought. We may have to just see what the hell happens with RFK Jr. and with Dr. Oz. I mean, I don't know where I am in the time-space continuum to be sharing optimism in the context of all these shifts, but we really will have to sort of navigate some structural shifts, I think, because I don't see Black people in the United States, or Latino people, whose abuelas and tios are being deported in chains, and by the way, live streamed on TikTok, in case you're interested in tuning in. I just don't see how these people are going to say, oh yeah, now's the time for me to sign up for that government-sponsored clinical trial with Pfizer, So I'm just trying to be a little bit more realistic about some of the structural headwinds, but also structural radical opportunities that lie ahead.
So Nick, I want to follow up on your point about radical changes. I think the GLP-1 story narrative absolutely shows us a path in terms of how a medicine can be preventative and not curative, that's not the word I'm looking for, but it has broad impact in overall health state of an individual and the populations. And, from a productivity point of view, there's just no comparison of the ability of that one pill, or not pill, that one injection, medicine, in terms of increasing productivity of healthcare versus the traditional way that we try to change behavior and provide services to, address ailing populations. So I think it is a great example of how we can do radically differently. And that get me to this increasing interest and effort in health span, not longevity per se, because that suggests us living longer, but health span research and medicines. And I think there are huge opportunity there to go after. But I, want to bring it back. In order for us to be effective in delivering the next generations of health span medicine, we're going to need to have better access to the patient populations and better access to the data so that we can have better surrogate biomarker, because we don't want to run a trial for 50 years. I mean, that is not realistic, and it will be hard for your investor like you, Nick, to even, commit that kind of resource. So how do we make sure that we are developing the right measurement or metric, Danny? Because if we don't, then we're going to have a generation of medicine that optimize for something but in the end, not really the health span of the society. And so this is the time to make sure we do that right, because this is the beginning. And I do believe that we have to take into account the heterogeneity of the populations. That means the diversity, including, certainly the understudy, what I call the invisible 80%, of the population. Because if we don't have data on them, we won't have surrogate biomarker that measure the impact of this medicine on them. And the industry will be optimizing for the medicine that will improve the other 20% in whatever the measurement is. And so the one radical idea that, I've been thinking, and we had this conversation at lunch, Nick, is how do we incentivize patients to participate and contribute their data to research and clinical trial? Not so much based on trust, because you just make a good point about, can we count on trust, but based on economic return. Why on the data producer, which are the patients, or the individual who contribute the patient, why are they not in the value chain of any kind of medicine that get developed and ultimately go to market?
Well, that system is different, Linda, in other countries. Other countries in the EU have started to, for publicly sponsored or federally funded activities, including drug reviews, clinical trials, and the like, they have public benefit subsidies, or they have a structure where you are capturing some of that value. The United States, being such a financialized economy, has really failed to do any of that value capture, and this is really the legacy of the great hoax of society from the 1980s of trickle-down economics, that if you pad the pockets of executives and rich shareholders, that will trickle down. But as we know, it actually trickles up. So these are radical ideas. Some of these ideas may be actionable if we hold this current administration to some of the wild things, radical things that they do say from time to time. Like today, Donald Trump saying that some of the savings that our good compatriot, comrade, Elon Musk from South Africa, that beautiful bastion of democracy and liberty and equality, that some of those savings can be returned, actually shared to taxpayers. First of all, I'll believe it when I see it, but maybe we should take this idea and run with it. Maybe this should be something that that is emphatically requested of the federal government, that we actually return some of this public benefit, and that we capture some of the economic activity that taxpayer dollars go. Look, the friendly American taxpayer has been the cash cow of innovation around the world for a long time. I don't believe in deglobalization as a viable economic strategy, because I believe in facts and information. But I do believe that the American public deserves to have a little bit of a return, other than just the fact that we get cheaper items from Temu and Shein, which I've never shopped at, by the way.
So a lot of interesting topics. Also, I realized so far the bias has been toward drug discovery of a disease, of a target. And I'm glad we brought up prevention, because that's, of course, even better if nobody gets sick and they have a long health span throughout. So that's another thing we want to keep in mind as we
But I want to
breakout
make the
sessions.
point, but we're still talking about targeting, developing medicine against a target.
Right. Exactly. It's still the old paradigm. So I would challenge us, especially this afternoon in the breakout sessions, to think beyond that paradigm also, and how the technological advances in AI and other methods could potentially get us out of that paradigm. So,
well, I've been really encouraged by some of the data I've seen where, using longitudinal data, molecular data and clinical data, and sort of observational data that allow people to project and build a model to project how someone is going to do with a certain measurement that we agree on, like cholesterol or BMI or, and so on. And then within that model, being able to modify a parameter and ask what the impact is on that. So in that context, there's definitely going to be behavioral lever that you can pull. But I think there will be by necessity, well, not necessity. I believe that there will be the GLP-1-like target that we have to hit to improve the health span, because there is that natural heterogeneity, even in the natural population of human beings, in terms of their health and their longevity and their health. So being able to prevent disease in the subpopulation that has a certain tendency to develop disease means we have to understand the mechanism, the aberrations, and have the molecule to correct that, so that they can live a healthy lifespan like the next person who may, have less of that susceptibility based on their genetics and their environment. So I think it is not that different from us thinking about developing precision medicine, but in this case, it's about precision prevention, because I'm pretty sure we don't have one strategy that we can all follow and we all be happy. I mean, healthy, because I think it's going to be different for different individuals living in different part of the world, having different diet, doing different job, and having different exposure.
Yeah, and what a healthy diet is in each of these different areas. So James, you have been very patient so could you please go ahead?
Oh yeah, it's a slightly different question, but along the line, we talked about the limitations of the data, and we also talked about the potential of the system collapse and opportunity to rebuild. But in either case, in a hypothetical scenario where you can rebuild or you can recreate this and doing it the right way, what are maybe one or two things you would do a little bit differently so that the type of data, the type of infrastructure will be more helpful and more productive?
Let's start with that.
I'll defer for a bit. I want to hear what other people say, but I agree. It's a great question.
Linda, do you have any immediate thoughts of what this would be different?
Go first.
Okay, Nick, online, what do you think?
Uh-oh. Can you repeat the question?
Yes.
Great question, because everyone's punning.
James, you want to repeat your question?
Hit me, James.
Yeah. So the question is basically, if you had the opportunity to rebuild the system, what would you do? What would be one or two things you would do differently in terms of different way of collecting data, different type of data, or different infrastructure that will make it more productive and more useful?
I love this. I love this, James. So so here's the thing. Look, I am not a bystander of history. You know what I mean? And you sound like someone who's not either, James. I don't want to just rant. I'm very good at ranting, by the way. I don't know if you've caught on to this, but it just seems so, I don't know, so unsatisfying at the end of the day when I go to sleep, and I do my skincare and I'm like, oh, well, didn't really achieve much, but, and my vocal folds are, stressed. So instead of just ranting, I've thought about this sort of radical embrace of the fact that most of what we know in terms of academic and clinical enterprise is probably up in the air. I'm just sort of following the macroeconomic sort of bouncing ball here. What would I do differently? Well, I wouldn't say that appointing a 25-year heroin user as the head of HHS would have been the top thing on my list of to-dos. But since this is where we're at, someone who is interested in preventative health, well, if he's interested, as he says, in a science-based approach to addressing or enabling that sort of happy-go-lucky, blonde girl frolicking, like, Blake Lively frolicking in a field of wheat, sort of vision of health, right, and prosperity, what if we help to work with him to inform him on the role of science in longitudinal tracking of patient data? What if every taxpayer were given, or every member of Social Security, every contributor of Social Security, were given the opportunity to actually participate in longitudinal study? Now, some of the skeptics have really been afraid of the potential of surveillance, biosurveillance, but there's no way to get around the need to truly take precursors of patients' health. By the way, this guy RFK, who's not my favorite scientist in the world, I'll say that, but these are things that he has said and mentioned. And I think so much of the attention of our attention gets sort of invested into the obvious shock, right, of some of these appointments, that a lot of the leadership currently in these agencies lack the agility to navigate some of these changes. To your other points of like, what would you do differently? Well, I'll get back to you on that one. We have so many changes in this administration across federal agencies. But I'll be honest, I don't know what I'm even, how would you say? I don't even know what I'm fighting to preserve anymore because of the lack of receptiveness of federal agency leadership. You know, folks have, my calls, for example, with the Department of Defense and with the NIH and with NSF this week have focused on people basically telling me, you don't know what the hell you're doing and you're an idiot, which, by the way, I've been told that by much better, right? I've been called worse by much better, right? One of the things is, look, if people want to slash budgets for sort of clinical and academic or scientific enterprise, what if we actually took them at their word? And what if instead of just sort of throwing out the baby with the bathwater, by the way, I do not believe that we need less scientists. I think we need more scientists, but we need more scientists who are, how would I say, who are reliably employed where they can actually follow ideas to their natural end. The current system and the current structure of our economy just does not allow for that, just to be very clear. Most scientists struggle really bad. I would actually think about changing that. What if, thinking about the scale of investment CapEx, that what do they call it, Sanmi? The Magic Seven, the Fabulous Seven, something like that. The big tech companies, right? They've all had a sort of a pipe dream of being able to come in, swoop in with a little, tech here, a little efficiency there, a sprinkle of magic, and bada bing, bada boom, you got some healthcare, America, modernized healthcare systems. Well, it hasn't worked at either Amazon, JP Morgan, and the likes. But what if we actually invested in privatizing some of these, the scientific endeavor in the United States, and modernizing scientific enterprise across even academic institutions that are federally subsidized? What if we did that at the scale of CapEx into AI infrastructure? Okay, maybe $100 billion per year might be crazy. It is truly crazy for AI infrastructure. But what if we did half of that? That's $50 billion. That's the entire NIH budget per year. What might be possible by doubling the NIH's capacity, NSF's capacity through private enterprise? I don't know, quite honestly. And I don't fully understand how to sort of keep folks honest. But if we thought about having the American public as the customer, right? And if we thought about having the American public also as a shareholder through the federal government, some pretty disruptive and radical things might be possible in terms of modernizing our approaches. And some of those approaches might actually incentivize some of this preventative health of which we're sort of talking around in the abstract, we have to talk about it in the abstract, because to be very clear, when I say radical, this is it's not a monotonic. This is an absolute 180-degree departure from the way that healthcare is subsidized and financialized today. And the possibilities defy my imagination, to be very transparent. If that might actually dovetail with AI as a capability, but not a means to an end, which, will take a little bit of work there, to be clear, structural work. I think we might have options. We might be, as my grandmother used to say, cooking with gas.
So we have one more question from the audience.
Let me
do both of you have a
give it a try.
Yeah.
Okay. I think I going along the radical, I'm going to skip over the current administration and what we can and cannot do given the current administration to answer James' question, because that wasn't your question. Your question was, if we were able to rebuild, what would we do differently? And I would want to see a system where our care delivery system is more intimately connected to our discovery engines, so that there is the right structure to allow us to actually learn and capture data from every individual before and after they are patients and during the time that they are patients, because that really, we can't ignore the patient. I know we want to prevent disease, but it happens. So I think if we can have that system built where it is the standard that we track these individuals, and that there is whatever the structure to allow us to capture a sample that's for discovery, not just for care of the patient, because that's really the two umbrella that we fall under. And on top of that, create an incentive structure so that the patient or the individual that contribute their data and samples that ultimately lead to a product that has value, they should get a piece of the pie, no matter how small or how big, but they should be part of the value equations. And if there is no legacy system in place and you're able to build that from the ground up, that's actually doable. What makes this hard is going against the existing system. So that would be one benefit. If the system collapsed, we have to rebuild, I would want to build something like that, so that now from birth to death and sickness and wellness, we are able to capture data from individual and use this as a way to proactively intervene when they go off the track of health, so that's your preventive medicine, but develop the right kind of medicine that is individualized for them when they get sick, so that overall we can extend the health span. I'm not talking about us living longer, because that introduces a whole new different challenge to the society if we're all going to live to 150. But just be healthy rather than needing 3 caretakers because you were sick for the last 30 years of your life, which our society cannot afford. Then how do we do it so that we are productive and healthy until the last day? And I think that's the way to do it. But there are privacy concerns, incentive concerns, but I don't believe either one of that is technically impossible, but it is impossible in the current system.
Tammy.
Yeah, I think it's very well said by Lynn and Nick. I don't think I have much on the substantive side. I do have a question that I might frame around this. So I think I heard sort of data collection being incentivized better. I heard about sort of incentive alignment around institutions and broad society, such that, I guess to the earlier comments, we're asking more of the right question and framing the research enterprise in such a way that it productively impacts society. And then I think one that we hadn't talked about as much, but resonates with me personally, is thinking about how we support higher risk research, and so people taking chances to have sort of much more meaningful impact. On the data collection side, so strongly in favor of all these, if to the earlier point, if we had an opportunity to redo it again. And we may or may not, as we might see. But on this question of the data collection, and this I ask from a position of curiosity more than anything else. So my understanding is that some of the incentivization aspects of these things and then the sort of data collection parts of these things are tried in parts of, say, Europe. And I don't know what the success of these things are in those environments. And I'm curious if anyone, I guess, other folks on the panel or folks in the room know. And again, I say this from these sound right to me, but I'm curious if there are some natural experiments that we could learn from. So do we know when, to what extent have these been tried and where have they worked or not worked?
So we have one last question from the audience from Dr. Burston to close out this panel session.
So to address to ask a question about from all of you, as someone who represents the funding agencies, and you're talking about sort of several things that I heard. One is, opportunities for transformative approaches, which maybe aren't actually being supported. The fact that a lot of what gets done may be driven by the data that's there, right? Rather than the data we might like to have. And also that there's, I would say the incentive system works against robust data collection and curation. What suggestions do you have for those of us who are in the funding agencies to think about how can we better support a ecosystem that's going to actually, so we talk about the goal of supporting transformative science, and I'm afraid that that word has very little meaning in the context of the way we support, and that's what you're saying.
I agree. I think your last point about high-risk research, I think the funding mechanism, a structure, is actively disincentivizing high-risk research. It comes back to the incentive system, the fact that you have to get past peer review, you have to get publications, and high-risk research, by definition, need to fail. Otherwise, you're not doing anything high risk. So, where's your safety net? Because nobody wants to publish a failure. At least that's not the publication norm. And you don't get promotions, and you won't get the next grant, right? And on top of that, even getting past peer review, since I'm no longer in academia, I can say this, they're not peer review because people who end up serving on the study section often are not the people that are really the right peers. I don't know why. Not everybody has to. I mean, why is it that there isn't a requirement for anyone who gets funded to contribute back and serve in study sections, right? And, I've seen so many times and personally, experienced it where my resubmission finally get funded after I completed all the aims and prove that all the aims can be done because I've completed it. And now my grant's funded. So now I can do the next thing. That is fundamentally what's wrong with our current system. I don't have an answer. I don't know how the peer review system can be improved, but it certainly does not incentivize transformative research.
Wait, can the questioner, can the person in the audience reveal the funding agency?
We all know it's
So I'm director of chemistry at NSF.
Oh, wonderful. I like chemistry.
How would you like what could NSF or NIH or these different funding agencies change the incentive system? Because you've also mentioned the perverse incentives that drive us not to doing the most important work, or the collecting the data that can be easily collected versus the hard data that needs to be collected to advance the problem.
Yeah, so I think these issues come up over and over again in many of the decision-making processes that I get to see. I think many of us get to see, in a somewhat narrow sense, this comes up in faculty hiring. Same thing comes up in, we just finished student admissions at Stanford. We want to be taking chances on people that could be amazing, but haven't shown it yet. But we're actually extremely defensive in practice and are looking to actually minimize false positives much more than actually optimize for the chance of a good outcome with the risk of maybe not. So you end up picking people that pattern match to essentially do. People that come into PhD probably should already have at least Stanford CS PhD. Like these are extremely accomplished people, but we're not taking chances there at all. And so, I mean, I reflect on this a ton in those situations. I mean, it's unclear to me that anyone is, I guess, doesn't recognize the problem. I think we all see it, or at least most people I talk to resonate. Even people who, in fact, I think all of us who are successful have been successful because we played the game as it's currently expected. So we do some high risk stuff, but like bounded very narrowly between stuff that people will recognize as likely successful. I think Lynn, as you so well said, so you essentially do the next thing and then talk about it afterwards. What is the mechanism to incentivize truly high risk, but still allow for? Because I think the backside of this is you also have to change the norms to allow for failures. Again, I think, as Linda, you already said, which my observation is politically, those failures are not handled well. So I guess all the way to also to this conversation about how we reflect back to society. Like, what is the version of the conversation where you say, we took some moonshots to make sure that, we may have the chance of curing cancer, but in doing this, we, out of 10, 8 failed. Like, what's the mechanism for going back to the taxpayer and saying, so this is part of what we're funding? I don't think I have answers. I could imagine, so in terms of processes, I think we have some good ideas on how to do it. I think the backstop of execution, to my mind, is not knowledge of ways that we could do this. It's sort of how we sell this to purse strings, essentially, of this is the thing we're doing. And when I say we have mechanisms, my sense is, some of the mechanisms include, like, if people see that the reward structure is actually rewarding people who are taking huge, like, big chances, and they're getting a chance to try these things out, the community will realign. To my sense, it's community realign, and more people will take these big chances. It doesn't happen because everyone pattern matches to the observation that people who are successful are mostly not. And when they train their trainees, they say, don't take big chances, actually. Like, do the thing that's more likely to pass through grant review, or if you're in industry, get supported up or down the chain. There's a version of this in the AI community that's also very fun. All our models are actually the same thing. No one does anything different because it takes all the models are the same. So it takes a few $1 million or even more to train the big GPT-4 style thing. Other people see that this works. The chance that someone will try something that varies from that is actually very low. We're all doing small deltas on the main architecture, and like no one is really taking a big risk at the scale of sort of meaningful change. My sense is the problem is not, and this may be naive, so I'm curious to have further conversation, maybe in the sessions when we talk through things. My sense is not a mechanism like we don't know what to do. It's how do we sell it to people who will be skeptical of you're willing to take risk and have many failures to get a few successes. Some of this seems to me an education norm thing, possibly. Some of this could be trust. Some of these are tied to political whims that are, I guess, quite tricky to navigate. I don't know. I have a good answer.
We're actually out of time in
All
our
right.
session. Nick, do you have any quick addition of constructive thing
yeah.
that?
So I'm 33, right? 33 years old. And I just learned that I am not only a member of the largest generation in American history, but I am the prime demographic for podcasts. So take this recommendation with a grain of salt, but we have a communications issue. I got into science because I was absolutely dumbfounded as a teenager, maybe when I was 13, 14, by discovering this biological phenomenon called quorum sensing. So Bonnie Bassler, Johns Hopkins, I can't remember, she's at Princeton. I think she's at Princeton. But quorum sensing, where you basically have these programs of bacteria these hyperstructures of bacteria that all talk to each other based on their cellular density, their colony density, and diversity. And then they regulate based on that to sort of like bifurcate into little projects, if you will. And I think about funding agencies and funding ideas in areas in that way. I will say that ARPA-H, right, which is sort of certainly up in the air right now, but ARPA-H has taken this general approach of greater programs that break down into projects into account. The problem is that those projects never talk to each other. The way that researchers talk is they go to NeurIPS or they go to Gordon, GRCs, if you're a chemist, sometimes in fabulous locations. But the big problem is that the connectivity of these ideas and the reciprocity of these ideas and thinking about leveraging multiple projects across multiple institutions and multiple research labs as delevering systems, right? As a way to sort of, how would I say, to take advantage of the logarithmic rule that venture capitalists get to take and investors in general get to take of all the time, which is that 1 out of 10 will return the losses of the other 9. I'm curious if this approach might be tractable in federal funding agencies. Today, from the current sort of structure and the limitations and that, believe me, I understand the handcuffs placed by Congress, the answer is definitively no. But if we think a little bit on the along the line of radicalization of the scientific or modernization, modernization and transformation of America's scientific enterprise, there might be something there. Why should the research of one person in one institution that just may not have the equipment or the institutional support or the overhead or the indirect funding, yada, 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 why should that one not be able to sort of collaborate by structure, by design? And why shouldn't the winnings be shared? Another reason that I'm interested in that model for approaching sort of the finance, the structural side of federally subsidized research in the United States is that I just can't keep up. I was always known as the guy who could read all the papers, and I can't read enough paper. I read papers all the time, but I'm overwhelmed, and my AI can't do it either. So I need more impact through quorum sensing. And people can Google that if they want to find sort of the Cliff Notes description of what that is. But I think there are different approaches that Congress, probably out of necessity, will start to increasingly embrace that will, I think, maybe better feed the national interest. How we actually address the challenges structurally and tenure and promotion, that's a very, very different topic, but we're going to have to change things. That's for damn sure.
With that note, what I'm hearing here is a little bit that AI can also inspire us in the sense that AI so-called negative data is not actually negative. It's actually something that allows us to get to where we need. And it's also extremely dependent on lots and lots of individual small pieces of data that your latest X message or LinkedIn post or whatever that fed this algorithm didn't seem very significant on its own, but then you start bringing it together in a way that's much bigger than the sum of the parts. And then we need to think those of you who are here will be going to we'll take a 10-minute break and then come back to start thinking about these things as the work in the workshop. And for those of you who have joined us online, we hope you'll join us again tomorrow to continue with some of these fascinating discussions and going on and thinking about how we go forward. So with that, join me in thanking the 3 panelists for fascinating discussion.
Thank you.
sticking around. There is sugar and caffeine to get us through this, so stock up, and I'm excited about this last panel. Well, thank you. This is our last panel for the day. What the impetus behind this was Apoorva Dave, who is sitting up there, is my colleague, and asked me to join a foresight activity for the Climate Security Roundtable that he organized. And Laurel Royer suggested, hey, let's have a breakout room for this. You know, let's hear from the audience, the attendees. Like let's hear from them. What would be a successful breakout room? I contacted Apoorva and like, well, let's try a foresight activity. So we pestered Aaron Carmel to come and join us from American University, and Apoorva. I'm just going to hand it off to Apoorva and Aaron and Laurel to just kind of walk us through this next and final session for the day. And I really liked how Nikki reminded us this will be the work and workshop. So thank you. There's a participation grade later.
Good afternoon, everyone. How y'all feeling? Ready for some additional brain gymnastics? Excellent. Good professors. So good afternoon again. Apoorva Dave is director of the Climate Security Roundtable here at NASM, and he will be having just a very great conversation, very brief, so that we can head out into our small groups and breakout session. He'll be having this brief conversation with Dr. Aaron Carmel, who's professor of information technology and analytics at the American University, and Aaron is considered a futurist, right? Excellent. And then Apoorva will guide us into our breakout session, the activity, how it's planned. But first, we'll just have a little facilitated discussion, and then we'll chat a little bit about what's next.
Great. Thank you, Laurel. So hi, everybody. I'm Apoorva Dave. What you have in front of you today is an oceanographer and a futurist who are going to try to walk you through a futures exercise in all of this. So it's been a really illuminating conversation for us. And I guess I'll just start off by saying that what we're about to do today is meant to be an opportunity for creativity, an opportunity for cross-linking across your different perspectives. It's not meant to be, I think, like a rigorous kind of formalized, structured activity. And I'm sure that many of you have engaged in this type of exercise before. But what this is really meant to do is to be an opportunity to explore an uncertain future, which we'll be talking about how, why we're doing it the way that we are, what's the potential value, and what's the best way to go about doing it. So, Kay, would you mind going to the first slide, please? So my job for the first 5 minutes is just to walk you through what we're going to do, because I think there's a lot of value in kind of initializing and orienting the group as precisely as possible before we do these exercises. So the exercise that we're going to do today is modeled after an activity that we've done in the past with our Climate Security Roundtable and other activities that we do with our national security community partners. And again, I work a lot with the intelligence community, and this is a community that has many, many different methodologies for structured analysis and kind of exploration of uncertain futures. And this is not an attempt to kind of do a wholesale tabletop or anything really that formal, but it is an attempt to kind of resurrect some of the things we've done in our workshops for this group that I think have been useful and generative of interesting ideas. So the the basic directions for this group are that we are going to break you up into groups, and the final slide in this deck will show you where you're supposed to go. We have 3 rooms, this one and 2 other rooms, which you'll be directed to. Each room will have somebody on staff to support you. But once you arrive at those rooms, you will be in a small group. We'll ask you to identify a discussion lead and or a rapporteur. It could be the same person if you want. You structure it however you like. And the rapporteur will be responsible for kind of creating a record of the conversation, which is important because we're going to be coming back in plenary to synthesize those discussions that happen in groups. And then I can't read this. Oh yeah, okay. Oh yeah, I have it right here, sorry. So what we're going to ask you to do is then each group is going to be assigned a scenario of a plausible future that exists in the space. One will be positive, one will be negative, one will be kind of a middle of the road, business as usual type scenario, and we'll explore what those mean in a little bit. And to respond to the discussion prompts that are on your sheets. And every group will be given a set of written descriptions for your scenario, and everybody should have their own individual copy. Everybody should also have an individual copy of a discussion sheet with the directions and the discussion prompts that are written out in some more detail than what's here. But the discussion then really will entail kind of you're responding to three sets of questions. So the first thing is we'll ask you as a group to consider your scenario, to refine it in whatever way you think is necessary, to add nuance, to make revisions to kind of the way in which we've described it, and also to provide maybe some of the overarching societal and or social and cultural and economic and political and governance kind of context that underlie that scenario. And then the idea is that we will have described a future for you. And then the second prompt will ask you to place yourselves in that future. The time horizon will be the year 2050, because that's the one that the CSR has chosen as kind of its, midpoint in the century, an opportunity to take stock of, of the trajectory that we're charting. And obviously that's a midpoint that resonates with a lot of different communities, right, that are thinking globally. And so basically to place yourself.
But that connects to one studios, and in the Hollywood studios, what do? They do scenarios. And that's where the term we want you to go here. So that's a little bit of, just rattle them off. The 5 Ps are plausible, possible, preposterous. We're going to leave preposterous out of this schedule here. One of the important ones is probable is the one that we rational actors think is the most likely. Another name for it is, now that's very important, or another name for it is and probable. Now, that's very important because we have to set a baseline for ourselves as we think about, anything.
And so for policy customers in the national security space, it's really a question of just like more likely than not, 50% one way or the other, and they're tremendously consequential choices that are made from these, whereas in other spaces, maybe a greater amount of certainty is required. And so my question to you is, given the fact that this is kind of a hand-wavy exercise, the way that we're doing it, what is the value of doing a foresight exercise, and what do people actually get out of it? When she was kind of thinking about this activity before, was that this is, in addition to an opportunity to kind of be imaginative and generative in our thoughts about how the future is going to go, it's an opportunity to cross-link and network.
Bringing here, in me sitting here as a futurist and using futurist terminology is opening up your mind to a different way of thinking than you usually do. The very paper-based, structured planning methods, we'll set them aside by and large and really open up your creativity here. One of the key things in this exercise that you'll do this afternoon is have fun with it. Laugh, enjoy. That brings out the creativity in you.
Great. Thanks so much, Aaron. It's kind of obvious. It's asking people who've sat in a room all day to get up and move to other small rooms where they can just loosen up and have an informal conversation with colleagues. I don't think we need to convince you that there's value in that. So we'll go ahead and do that. But
what if the audience has some questions?
Yeah, I was going to ask Laurel if you had any kind of things that you pulled away from the discussion or thoughts you wanted to share. If you have questions, we'd be happy to take them.
Thank you very much. Very insightful. Any questions before we get started? All right. And yes, Nico.
We have a readout to do tomorrow.
Yeah,
Yeah.
sorry.
Yeah, explain a little bit more about, because we're gonna guarantee to have great discussions.
Yeah.
But then what do we have to, then there'll be panic because we need to turn in the assignment.
Yeah, there will be. Sorry. Yeah, great question. And this is always where it kind of falls apart if it ever does. So Kay, can we look at the last slide? So this is these are the group assignment, or sorry, these are the group assignments. This is where you're going to go. Once you get into groups, you're obviously select a lead, select somebody who's going to take responsibility for kind of taking notes and synthesizing the discussion. And then when you come back tomorrow, the plenary synthesis can be whatever you decide to make it. So if you fail to exercise your imaginations, it will end up being a readout where one person gets up and you read out your notes and then the next person, if you do it well, then there will be somebody, I'm not going to be here, Aaron, and I will not be here, but somebody could lead this as kind of a seminar style discussion where you give people a chance to kind of, read out. We keep those readouts short. And then you really actually don't just go around the room and let people talk, but somebody actively manages the discussion and follows a thread to the end and, hopscotches around the room, but really tries to pull out like the similarities and contrasts. And the expert should actually tell us how to process them.
All right, I'll challenge the groups. One of the things we do as futurists is in the readouts is we actually do skits. So those of you who are brave, you can design a small skit to illustrate this future, bring Halloween costumes, whatever. You can take this further.
This futures exercise that I ever did was, we were at this weird retreat that was funded by some private foundation in like a monastery somewhere. And we did this futures exercise where we did exactly this as Hindcast. And every group came up and it was basically two people pretending they were in the future, having a conversation saying, do you remember the last 15 years and what it took to get here? Or like, this is a really crappy world, or this is a great world, or how do we just keep doing the same things that we, we're doing? And then you just kind of, you could play act and think of, that was really fun. So.
So a couple of things. Not only are we asking you to provide us input and insight for this workshop, but I think Marty and Nicola will agree that this information will help inform the planning of the CSR moving forward. And so you leave your mark with us beyond just this session and allows us to continue to build, and the conversation doesn't just stop after tomorrow. So please, get out of your lane. This is your opportunity to drive a little bit crazy and engage. And so before we get in our rooms, I'm going to ask you, because, there's the saying that say, you don't always remember what people say, but you remember how they make you feel. So I'm going to ask you to do this quick little thing with me before we move on. So if you guys could all stand. Andrew is looking at me like, where are we going with this, Laurel? All right. So I'm going to ask you to just fold your arms. That was super easy, right? Like, how many of us had to think about how we're folding our arms? Nobody. Okay, so unfold your arms. And I'm going to ask you to fold your arms, putting the opposite, right? Like like put the other arm, right? So this time it wasn't as comfortable, right? You had to stop and think a little bit. And it's like, wait, like I can fold my arms. So this is sort of like the exercise that we're asking you to participate in. It might be a little uncomfortable. You might have to stop and think a little. You might have to say, wait, what? I know this, but what is the question? So thank you very much. And we can ask you to go through your poll. Yeah.